Hello future doctors out there, I hope you're doing great my dear children, and as promised we are bringing the previous year paper solution for the 2024 year, and this time I'll be talking about zoology solutions. So all the animal lovers assemble right over here, and I'll be making sure that you're getting 180 on 180 in zoology. So those of you who have already attended the paper, you were not able to solve the questions, don't be worried. I am going to give you in-depth solution for all the questions. I'm going to make you understand the concepts around it such that if you get another question just like this, you could use that concept and solve it properly. So bear with me throughout the entire video, and if you're someone who have not yet subscribed to the channel, do it right now. Okay, hit the subscribe button and also tell it to your friends that hey, there's a NEET channel. I think you should be looking forward to it. Yes, do that. Okay, do that for us. And also one more thing, if you are someone who have not started practicing questions, okay, along with your concept learning, then it's high time to do so. Okay, so after every 3 to 4 days, practice one mock test. It will give you a real-time idea of where do you stand and what is your speed and accuracy. Okay, with speed you have to also calculate the accuracy. So there can be people who can go like this, but how accurate are they? You have to analyze that as well, right? So please do that. Please take mock test. If you're someone who's finding difficulty in finding the correct mock test, we have something for you, and that is totally free. Okay, there is no, it's not charged at all. It's totally free mock test. Only there are 5 of them. Please make utilization of these 5 mock tests if you are someone who is finding difficulty in finding a good mock test. Remember, these mock tests are being made by teachers who have been teaching for NEET for years. So we have a 25 plus years of legacy of creating NEET champions and JEE toppers and everyone out there. So trust us and you can utilize this. If you find there's something else useful, you can go for that as well. It's your wish, but the link to this will be given in the description box. Try it out once, then only you'll get to know, right? Yes, and do not try difficult question paper from any places. You know, NEET is not just about difficult questions. It's a mix of easy, moderate and difficult questions, and difficult questions are only 14%. Okay, always remember that. Okay, so some people may give you what's say difficult mock test, and it might demotivate you. I do not want you to go through that. So choose the correct mock test while attempting it. Going ahead with the first question that we are having over here, which of the following is not a steroid hormone? You have to rule out which is not a steroid hormone. So cortisol, testosterone, progesterone, all of them are basically steroid hormones, my dear children. There's only one over here, that is glucagon, which is actually not steroidal, but it is a peptide hormone. Okay, it's what? A peptide hormone. It's a proteinaceous hormone, but not a steroidal hormone. So wherever you see all this cortisol or basically the sex hormones, most of them would be what? Steroidal hormones. Always remember. So testosterone, progesterone, they are sex hormones. Okay, now cortisol, it's produced by adrenal glands. That is also a steroid hormone. Glucagon, insulin, okay, glucagon, insulin, all of them basically they are peptide hormones. Okay, and what is the function of glucagon? It breaks down the glycogen, okay, it breaks down the glycogen back to glucose when our body needs it. Always remember. So with this, I gave you 3 concepts to remember. What is the function of glucagon? Also that they both are peptide hormones and all of them are steroid hormones. So please keep that in your head. And children, if you're enjoying and if you think you want to score 180 on 180, then put it in the comment section right away. I am gonna score 180 on 180. Okay, you are going to comment that in the comment section below if you think you can. Okay, only if you think you can, then only comment. Okay, otherwise no need it. Now, given below are 2 statements. Statement 1 says, the presence or absence of hymen is not a reliable indicator of virginity. Is it true or false? So there is hymen, okay. Inside the vaginal tract, there is a very small membrane out there which is known as hymen. So some people, okay, have their hymen tore, okay, it would be broken, and in some it may still persist. Okay, now can I say the presence or absence of hymen is not a reliable indicator of virginity? Yes, can I say that? Yes, it is not a reliable indicator. Why do I say so? Because, for example, some people do cycling. You might be doing cycling, or there might be horse riding, or there might be some intense physical activities where what will happen? Where the hymen could be easily tore. Okay, the hymen could be easily tore. So, can I say that they have lost their virginity? No. It's not a reliable indicator for that. So this particular statement is absolutely correct. The presence or absence of hymen is not a reliable indicator of virginity. This is true. Okay, and Should someone be bothered about that? No, obviously no. Okay, now statement 2. The hymen is torn during the first coitus only. The hymen is torn during the first coitus only. I just told you right now, it is not a reliable indicator. That means it can be torn anytime during any physical intense activities that are being done, my dear children. So this statement is false. It is torn during first coitus only. Wrong statement. So this is false and that would be true. And this particular concept have been asked for so many times in different examination. Please make sure you know that. Okay, so I'll say statement 1 is true, statement 2 is false. Statement 1 is true and statement 2 is false. This would be the answer. I hope you got it as well. Okay, now next question. Which of the following statement is incorrect, or basically are incorrect? Let's see. Is incorrect? Statement is incorrect. Okay, a bioreactor provides optimal growth condition for achieving the desired product. You know, bioreactor, they are very big vessels, my dear children, where the small microbes are grown. And obviously to grow them, obviously you will provide them food, the culture and everything, optimum pH, optimum temperature, oxygen, everything. So obviously that is true. Optimal growth conditions are provided because we want them to grow, because they are going to produce our products, whatever proteins and everything what we want. They are going to do it. So that's why we'll take care of them. Okay, we are selfish people, but yeah. So most commonly used bioreactors are of stirring type. Are of stirring type as in stirred tank bioreactors, where, my dear children, there are impellers like this. And what do? They run around. They run around. What is the use of this? Such that the oxygen and everything is equally mixed. So some bacteria should not cry that I am not getting enough amount of oxygen. In that case, they won't be working. So obviously, yeah, most commonly used bioreactors are of stirring type, and this is correct. Now the third one, bioreactors are used to produce small scale bacterial cultures. Do you think it is for small scale bacterial cultures? No, my dear. They're big, large vessels which I cannot even fit in over here in this particular diagram. So that is for large scale production, not for small scale production. If it's for small scale, probably I'll be doing in some lab, in some petri dish or things like that. So this would be a wrong statement, and we had to find what the incorrect statement. So this is the incorrect one. Now bioreactors have an agitator system, an oxygen delivery system, and a foam control system. Yes, all of it is there in the bioreactor. That is how it provides optimal growth conditions and things like that. So I'll say this is the only incorrect statement we have. Now I'll give you one tip while solving this type of questions. Okay, whenever you find incorrect and correct, underline or basically just think in your hand, you're talking about incorrect, so that it becomes more clear to you. Sometimes what we do is that we try to find the correct option. You know, that happens because we are in an exam like situation where we might be tensed. It's quite obvious to have it, so please have this in your head. Okay. Next, which one is the correct product of DNA dependent RNA polymerase to the given template? Okay, so basically you're talking about what is a product of DNA dependent what? RNA polymerase. So there is one RNA polymerase, my dear children, and this RNA polymerase is dependent on what? DNA. For what, ma'am? What does the RNA polymerase do? It will be making RNA, my dear child. It will be making RNA. And what is this process when you are basically putting all the information of DNA into RNA known as, it is known as transcription. It is known as transcription. So transcription happens in what direction? If you know, transcription happens in what direction? The RNA polymerase will be making a new strand from 5' through 3'. It will be making a new strand from 5' to 3' direction. So in which direction should be the DNA, because it is DNA dependent, right? So there will be a DNA strand, and it will make an exact copy of the DNA strand over here. Exact copy as in it will make normal base pairs out there. So this DNA strand should be in 3' to 5' direction. Then only it can work in 5' to 3' direction. Okay? Always remember, DNA will be in 3' to 5', but RNA polymerase will work in 5' to 3' direction. So let's see if all of them has 5' to 3' direction. Yes, all of them have. Now questions could be coming where they can give you exact RNA, but the direction could be wrong. So you have to be really careful for that. That's why I told all this thing to you. Okay, now we have the DNA given right over here. We have to make what? A RNA template out of it. That is basically going to be mRNA, right? So what are we going to do? We are going to write 5', and now T base pairs with A. What does A base pair with? A base pairs with T, ma'am. That is in DNA. In DNA you have T, but in RNA do you have Hymen? No. Instead of Hymen you have what? Uracil. You have uracil. So it will be U over here. Now C will be G. Okay. A will again go with U. T will go with A, G with C, G with C, C with G, A with U, A with U, A with U, and the list goes on. With this information, can we try to find our answer out? Yes. So AUG, AUG, UAC, UAC, CUAC, CGU, CGU, and UUU. Until this, it is correct. Let's try the next one. Okay. AUG. Now we can just match from here. AUG, UAA. This is wrong. Okay. AUG, UAC. Okay. UAC, CGU, CGU, and then just a second. Okay. CGU. And then UUU. Okay, so we have one more option like that. Let's go ahead. Again, this is whole different thing, so we have to cancel this and this out. Now let's go ahead. Okay, now T goes with A again, A with T again, sorry, A with U again, and T with A again, right? So next should be AUA. AUA. So here also it's AUA, here also it's AUA. Oh my God. Until now we have not found that out. Okay, fine, cool. Let's go ahead. Now GGU. GGU, here it's GGG, and that's how we spot the difference. So basically this becomes your answer. This becomes your answer. If you want to do it fully first and then try to find out, that also works. But in this way, when you do, my dear children, what happens is that you get things correct. Okay, in between, if at all certain things are mismatching, you might get things wrong, which is why I told you, do it halfway, and then check. It will be a lot more better for you to understand. So what will be your answer? This will be your answer. Now, okay, so this is the RNA of the templates. This will be the RNA that you get from this particular strand. Okay, what do you call this particular strand as of the DNA? You call this as template strand. You call this as template strand. Now, you are given this particular thing. Okay, suppose you are given this particular thing and you are asked to find the sequence of coding strand. You are just given this particular question, same question, and you are asked to find what is the coding strand. What does coding strand mean? Basically, when you talk about the DNA has two strands. One is the template strand that goes from 3' to 5' direction. Other one will be the coding strand. Does the RNA polymerase read the coding strand? No. The RNA polymerase does not read the coding strand. It's from 5' to 3' direction. It's the DNA. It's just the DNA. This is also a DNA strand. So template strand is given and you might be asked the DNA strand. So it will be what? Just the opposite of it. Same base pair. Instead of U, wherever you had U, it will be T only because you're talking about the DNA strand. Okay. Oh my God, something got activated. Yes. Got it? I hope you got it. So over here, you just have to remember RNA polymerase always works on 5' to 3' direction. That you have to make sure all of them are 5' to 3' direction. Then you have to go ahead, always replace wherever T is there U should be there. Okay, wherever T is there U should be there. That thing should be in your head. Okay, going ahead. Now, given below are two statements. One is labeled as assertion A and other is labeled as reason R. Assertion A says FSH, follicle stimulating hormone, acts upon the ovarian follicles in female. Does it act upon the ovarian follicles? Yes, it definitely acts upon the ovarian follicles, thus that they mature and things like that. Yeah. Now, and Leydig cells in male. Do they act on Leydig cells in male? FSH, until this is correct. But do they act on Leydig cells in male? My dear children, in Leydig cell, where is Leydig cells? In between the seminiferous tubules where the sperm are being formed. Okay, you have some space which is known as interstitial space. Here you will find interstitial cells, or what do you call them as? Leydig cells. What do? They produce androgens, the male sex hormones. They produce what? Androgens. But who are the ones that stimulate the Leydig cells to produce androgens? It is not FSH, it will be LH, luteinizing hormone. Always remember L for Leydig cell, L for luteinizing hormone. So this part of the statement is false. Leydig cells in male, wrong. This is wrong. This is correct. So whole statement becomes wrong because of that thing. Now reason are, growing ovarian follicles secrete estrogen in female. Does it? Does the follicles which are there, okay, FSH acts on the follicles so that they mature. The follicle cells which are there, do they release estrogen? Yes, they definitely release estrogen. Now, while interstitial cells secrete androgen in males, does the interstitial cell or Leydig cell secrete androgen in males? Yes, my dear children. And the most common androgen is what? Testosterone. The most common male sex hormone is what? Testosterone. So what becomes your answer, my dear children? This becomes your answer. This is correct. This is correct. This is false. So assertion is false and the reason is true. This becomes your answer. Okay? Works? Should be working. Yes. Now, in the question, if it was asked, FSH acts on the Sertoli cells, okay, and LH acts on the Leydig cells. Will that be correct? Here I have written FSH acts upon the ovarian follicles. Cool. Okay, that is in the case of females. But suppose in the case FSH, what does it act on? It will be acting on Sertoli cells, which are the ones that provide nourishment to the growing sperms. Okay, and whereas LH acts on the Leydig cells, as I told you. Always remember this. Always remember this. FSH has an S. Here Sertoli cells also have an S. Cool. Cool. Now let's talk about the next question. Match list 1 with list 2. Typhoid, what is the causative organism over here? Typhoid is caused by Salmonella typhi, which is what? A bacteria. Which is what? A bacteria. So this goes with, A goes with 4. Let's see. A goes with 4. There's only one option like that. Eliminate all the option and you get the answer. Leishmaniasis is caused by Leishmania. Yeah, Leishmania, which looks like this, if you remember. Okay, and basically there is only one flagella. Okay, and what? Leishmania is a protozoan. It is what? A protozoan. So this goes with this. B goes with 3, A goes with 4. Exactly, yes. Now ringworm disease, even though it's a ringworm disease, it's just, why it is known as ringworm disease? Because it forms rings sort of things on the body, my dear children. It's not a worm that causes this. Okay, it's a fungus. Microsporum. Okay, epidermophyton, trichophyton, epidermophyton and microsporum. Remember that. So they are fungus which cause it. So C1. Now filariasis, also known as elephantiasis, is caused by Vicariaria bancrofti or Vicariaria malai, which is a nematode, Aschelminthus, roundworm, whatever you want to say, you can say. Okay, so basically Aschelminthus, nematode, everything, okay, comes under this. So basically its filariasis is caused by a nematode. Remember that. Okay, cool. So this becomes your answer. Children, are you enjoying the session? If you are enjoying the session, do let me know in the comment section, and also hit like button. Very important. This is a growing channel. You know, we already have a channel as Deeksha Karnataka with lots of subscribers in there. All the great, wonderful children out there who helped us, okay, achieve that. Now we want this particular channel also to grow so that it reaches maximum people out there who are preparing for NEET, okay, who are finding it difficult to afford a lot of different things in their life. So we are here to help them. Initially, we will try our best to do so. So definitely make this particular video and also the channel reach out to different people out there if you think it's useful, only if you do so. Now, following are the stages of pathway for conduction of action potential through the heart. Through the heart, how does the action potential go on? So listen to me carefully, okay? So we have the atrium, left atrium, right atrium, okay? Now we have the right ventricle and the left ventricle, cool? Now what do we have over here? We have an SA node. That is the pacemaker. It starts off the action potential, spreads it out to different cells out over here. Okay, spreads it out to different cells out over here, and then slowly sends a signal over here to what? The AV node. To what? The AV node. AV node is like between the right and the right ventricle over here, more towards the upper side. Okay, more towards the upper side. Now from AV node it will go to the AV branches. Okay, there will be AV bundle that we have, and then from AV bundle it goes to what? Bundle branches. The bundle branches basically go, and here you have the Purkinje fibers. Here you would be having the Purkinje fibers. So what is happening? Basically when SA node is activated, here all the cells become activated and the atrium will be contracting. Sending the blood towards what? The left ventricle and the right ventricle. Here AV says that take a pause, okay, you pause, and basically what will happen? Slowly it will send signal to all throughout the ventricles. Now the ventricles would be what? Contracting when the Purkinje fibers and everything is activated. So this is how thing goes. So first should be SA node, that is the pacemaker of the heart. Always remember, this is what? The pacemaker of the heart. And this is a pace setter. Okay, it makes up the pace that hey, you have to do something, and this guy is the pace setter, as in it tells us slow down a bit. Okay, so this is the pace setter and this is the pacemaker of the heart. Remember that, and it is totally involuntary. Okay, it can get activated by itself. Remember that as well. So SA node E should be the first one. So I'll cancel this and this option because there is no E over here. Let's do proper calculation. Right now, SA node. After that we have AV node. After that we have AV node, right? So E and then C, okay, cool. And then AV node, AV bundle, yes. And then what? Bundle branches. So A and then D, yes, and then last you have Purkinje fibers. So this becomes your answer. Okay, so SABP, you can remember like that. SABP. I don't know how you can, but yes. Okay, works. A, B, C, D, G, J, Q. Okay, anyways. Now, next question. Match list 1 with list 2. So, subphases of prophase 1 is given over here, and this is a specific characters which are there. Okay, so what's up over here? So, dikinesis, and dikinesis, what happens? First of all, if you're asked about this question, you should know this thing. Lazy zebras paint doors dark. Okay, so this is the order in which it goes. So leptotene, zygotene, ficitine, liplotene and dikinesis. Listen to me carefully. What happens in each of the stages, I'm going to tell about that, so that you can solve any questions around it. So in leptotene stages, first thing, okay, the chromatin, everything, chromatin fibers which are there, it will start their condensation. It will start their condensation, as in it will try to form proper chromosome shaped structure. Initially they are just like this, right? So they want to make a proper chromosome like structure so that it can be divided equally, properly later on. So chromatin condensation starts off over there. Now in zygotine stage, what will happen? Basically, suppose I have one chromosome from my father, another chromosome from my mother in my body, in my cell. Now what do I call this? These chromosomes are known as homologous chromosome. What do I call them? Homologous chromosome. Why, my dear children? Because the genes over here that might be coding for height, I would also have similar genes over here that might be coding for height. Now these genes could be different from this, but both of them, my dear children, would be coding for height. So I am getting those two chromosomes which are the same genes out there, or same type, which might be coding for the same type of, or so basically height. If I talk about height, both of them would be having genes coding for height. Now one can be for tall height, one can be for dwarf height, but anyway, it's for height. I hope you understood that. So basically these guys are going to come together in the case of zygotine. What do I call it? 1 and 1. So this is known as univalent. When two of them comes together, I will call them bivalent. Now they are bivalent. And how many chromatids do we, how many sister chromatids do we have? So, or just chromatids, how many chromatids do we have? 1, 2, 3, 4. We have 4 chromatids. So I'll call this as tetrad. I'll call this as tetrad. So bivalent formation or tetrad formation happens over here in the case of zygotine. They are going to come together right over here. Does that work? Does that work? And also, how do they come together? Do they have something that will keep them close together? Yes. So there will be synaptonymal complex being formed. There should be some gum in between them, right? So this is the gum, that is synaptonymal complex being formed in the case of zygotine. Always remember, in this chromosome 1, 2, they are known as sister chromatids. They are known as sister chromatids. What is this for this? What is this for this? This will be non-sister chromatid. It's not sister chromatid, it's non-sister chromatid. Remember that also. Okay, now that also works. Okay, so now they are together. Okay, they have formed the bivalent or the tetrad, and this is what the synaptonymal complex. So all this thing happens in the case of zygotine. Now in pi-chitin stage, what happens? In pi-chitin stage, my dear children, there could be recombination nodules being formed. Recombination nodules as in something that will help them, exchange certain materials out there. That will help them exchange certain materials out there. So these are known as recombination nodules. So in pi-chitin stage, recombination nodules would be formed over there. Okay, and in diplotin stage, what will happen? Basically, in diplotin stage, these guys are gonna move away a bit. They are gonna move away a bit. So don't want to confuse you. Listen to me until now. I'll just quickly revise. Okay. In leptotene, the chromatin has started condensation. Okay. In zygotine, these guys, homologous chromosome came together with the help of synaptonymal complex. Okay. Now it's known as bivalent, or they're known as tetrad. In pi-chitin stage, the recombination nodules have come. The recombination nodules have come. With this limited amount of information, can I try to solve it as of now? Can I try? Okay, let me see. I'll talk about other stages as well, okay? But let me see if with this amount of information, can I try? So basically, leptotene is where chromosomes look like thin threads. Yes, they have just started the condensation. That's why they look like what? Thin threads, okay? Slowly at the end of prophase 1, they will look like thick ones, okay? Like proper chromosomes. Now, after leptotene comes what? LZP, Z. Zygotine. In zygotine, what will happen, my dear children? Appearance of recombination nodules? No, that happens in pi-chitin, right? So in zygotine stage, first, synaptonymal complex would be formed. Synaptonymal complex, the gum that is there, that will be formed. Now in pi-chitin, what will happen? As I told you, recombination nodules will be formed. What? Recombination nodules will be formed. So D goes with 2, 3. D goes with 3. I just have two options like that. I just have two options like that. Now going ahead, C goes with 1. C goes with 1, I just have one option like that, okay, and then B goes with 4, B goes with 4, yes, in the same option, and now A, what is left will be dikinesis, where what is happening, completion of terminalization of chiasmata. So I have not spoken about that, but still you got the answer. By just knowing about the 3 phases. You get the power of concept knowledge. With the knowledge, if you are having about concept, my dear, you can solve a lot with limited amount of information as well. But that's why we say you should at least know a little bit amount of concepts. So listen to me out carefully and do this. Now we'll go ahead with diplotin and dikinesis. What is going to happen over there? So I have written over here, if you want the PDF, you can comment down that you want the PDF. We will be providing the PDF as well. Yes. So basically in diplotin, if you see what is happening, the synaptonemal complex which is there, it is going to dissolve, my dear children. So ma'am, then the homologous chromosome, they will go far away from each other? No, because previously recombination has happened. You remember recombination nodules coming up? Yeah. So recombination, there's an exchange of things between them, and what happens is that basically these guys are going to remain attached at certain point. Okay, they are going to remain attached at certain point. They're going to exchange their stuff over here, and this is known as chiasmata. So in diplotin stage, breaking up is too hard, right? So synaptonemal complex dissolves, yeah, but chromosomes stay connected at the crossover points, yeah, forming what chiasmata, that is X-shaped structure. In some species like human, you say this phase lasts for years. So yes. Now coming to dikinesis, the last one. So what happens is that they still try to break up, but it's not happening. And when they try to break up, what is happening, my dear children? This particular chiasmata, it will move to the terminal position. When you do like this, what will happen? Slowly it will try to move down. So terminalization of chiasmata occurs in what? Dikinesis. Okay. So chiasmata move towards the chromosome end, that is terminalization of chiasmata. Chromosomes are fully condensed. By then chromosomes are fully condensed, and the meiotic spindles and everything appears also in this particular stage. So dikinesis is like do or die situation. It's the last thing. Always remember, die, the last thing, yeah, the terminal thing. So here the terminalization of chiasmata occurs, and over here the same thing is given, right? Completion of terminalization of chiasmata occurs in what? Dikinesis, and with that we solve the answer and we get the answer, that is 3. I hope you got it clearly. Now going ahead with the next question that we have over here, my dear children, match list 1 with list 2. Okay, this is the answer. I got scared. Yeah. 3 types of muscles are given, A, B and C. A, B and C. Okay. Identify the correct matching pair along with their location in the human body. So you have to tell what is this, and what is this. Okay. Okay, fine. The name of the muscle and location is given over here. So over here, B is like spindle shaped. It is what? It is spindle in shape, like this, almost like this. And if you see, it just has one nucleus. That is a clear signal that this particular muscle that you see, my dear children, it's a smooth muscle. Smooth muscle, okay. S starts with smooth, and S basically is for spindle shape or whatever you want to remember, and it always have one cell. And you know the good thing about smooth muscles, they do not listen to anyone. They are involuntary. They are what? Involuntary in function. They are involuntary in function. Now over here I have given certain mnemonics to remember. So S for smooth, S for slow, and S for self-working. So they are involuntary, they do their own work, okay. No one has to guide them as to what they have to do and things like that. And S stands for smooth, and they are unicellular. Always remember that. One cell with one nucleus. That is the very good thing about them, okay. So over here, if we have this information that smooth muscles, basically they are involuntary and they just have one nucleus, what can we solve out of it? So over here smooth is for stomach, and here smooth is for heart, and here nothing is given, okay, nothing is given. So we at least know that this is smooth, so I can eliminate these 2 options. Wherever I do not find smooth, I am going to cancel them. And I know B is smooth, so B is not smooth over here, so this is the wrong one, and B is smooth over here, so this could be a right one. This could be a right one, or this could be the only one that is right. But let's see. And my dear children, in heart, obviously you will be having cardiac cells. Ma'am, but you said smooth muscles are involuntary, so your heart muscles should also be smooth muscles, right? Cardiac muscles are there, they are also involuntary. Please remember that. Okay, so I can say smooth muscle in the stomach. In the stomach, there is what peristalsis is happening. There is mixing of things, Baba, right? Contraction, relaxation is also happening. So that, do you say that, hey stomach, you have to contract, you have to relax? Is it under your control? No, it is not under your control. That means it is also what? Involuntary. So smooth muscles are also involuntary. Cardiac muscles are also involuntary. But cardiac, as the name suggests, it's there in the heart. So cardiac will be going in the heart. So C structure that you see over here, it will be in the heart. So C for cardiac, right, and C also for connected. So if you see, these are the cells which are they are striated for sure. They have lines like this, and apart from that, they are connected as well. They are connected. So C for cardiac, C for connected, and C for constant contraction. There will be constant contraction. If it stops doing that, we are gonna die. Okay, and the last one that we have over here, it is what? It is just skeletal muscles. They are just skeletal muscles, and skeletal muscles, my dear children, they are also striated, but they are not branched. Okay, they are not connected. They are just skeletal muscles. They are striated, and they would be having more than one nucleus. They would be having more than one nucleus in a cell. So they are multinucleated. If you see over here, you can find basically, if this is a cell, this is a cell, basically they can have 1 to 2. Okay, in the case of cardiac cell, they can have 1 to 2 nucleus. So it is uninucleated or binucleated. But in the case of skeletal muscles, they are completely multinucleated and they do not have any connections. Okay, so S for strong and S for stripe. They have stripes. Over here also you have stripes. So in general, if you're confused, let me break everything down for you in just one particular line. Listen to me carefully. Okay. Skeletal muscles. Okay, skeletal muscles are striated. Okay, and they have more than one nucleus. They are multinucleated. Cool. They do not have any branches as well. And the good thing about skeletal muscles are they are voluntary. They are what? Voluntary. For example, I have muscles over here and I can do this. Why? Because they are voluntary. It's under my control. So your biceps, your triceps, everything comes under what? Skeletal muscles. Fine. Now, the second thing. So that's why strong, S for strong also. Now going ahead to the next thing, that is basically your smooth muscles. So smooth muscles, again, they are involuntary. They are spindle shaped and they are uninucleated. They are the only one that is uninucleated. So this particular smooth muscles are found in areas where there are involuntary functions happening. For example, your stomach, your things like that. Okay. Now talking about your heart. Your heart will obviously have exclusive things because that itself is exclusive. So you have cardiac muscles. Cardiac muscles are just like your skeletal muscles, but a little bit different. They are branched and they also have 1 to 2 nuclei per the cell, not more than that. Okay, that is the usual thing about them. Cool, works. So will you be remembering this? So skeletal muscles in the triceps, yeah, smooth muscles in the stomach, and cardiac in the heart. If you are able to spot this guy out, okay, because this is the unique thing. It has one whole beautiful cell with one nucleus, and that is smooth cell, my dear children. So if you are able to spot this, you can easily find the rest. Okay, so just because I spotted this, I got to know that B is this. Rest of the places, B was not smooth muscles, and hence I got to know this is the answer. Use your tactics. Yep. Now, match list 1 with list 2. Non-medicated IUDs, copper-releasing IUDs, hormone-releasing IUDs and implants. IUD stands for intrauterine devices, my dear children. So, non-medicated IUDs, as in if they are inserted inside the uterus, they are not going to release anything out there. They are not going to release anything out there. But then what is the use, ma'am? They're just going to act like a foreign particle. And when something foreign gets inside our body, what will our body do? Our body will try to fight back against it. And while it does that, okay, what will happen? Suppose coitus has happened. Sperm has been released. This sperm is also foreign, that will also be killed. So that is how non-medicated IUDs work. And in this, there is only one non-medicated IUD mentioned over here, and that is what? Non-medicated IUD, and that is lipase loop. Okay, now copper releasing IUD. Copper releasing IUD are the ones that will be releasing copper inside our body. What does this copper do? Listen to me carefully. Copper basically, suppose there's a sperm out there, okay? There's a sperm out there. Okay, the copper releasing IUD, the copper will be like holding the sperm's tail and saying, you cannot move. So it messes up with the mobility of the sperm. And if sperms are not able to move, can they go and fertilize the egg right over there? No, Baba. It cannot fertilize the egg right over there. So can I say copper releasing IUD over here? What do I understand? I'm not able to understand anything, ma'am. How do I understand this? So basically, I can say copper releasing IUD is what? Multi-load 375. Okay, so how many days do we have? 365. So just 10 more days you add, and that's how you get multi-load 375. So copper releasing IUDs work for like 365 days of an year, you can think like that. Okay, so multi-load 375. Apart from that, if you have copper T, if you have if you have Cu7, all of it basically are copper releasing IUDs. Fine. And now with this information, can I try to find our answer out? Let me see. So A goes with 3. A goes with 3. I just have 2 options like that, so I will be cancelling out these 2 options. Okay, this and this 2 option. Okay, now copper releasing IUD goes with 1, copper releasing B with 1, B with 1. I have 2 options like that, so I get to understand that I can eliminate 2, but I cannot find the answer. So I have to go again with the next one. Hormone releasing IUD. Hormone releasing IUD as in inside our body, if it is put, my dear children, it will be releasing hormones. And what's the worst part about hormones? Okay, what's the worst part about hormones? Basically, they are going to mess up around the normal cycle. So usually we know we have 2 hormones, progesterone and estrogen. Both of them are responsible for ovulation to happen. Both of them are responsible for implantation to happen and everything. But when their level increases way too much, they are going to do the opposite work. They are not going to do ovulation. They are not going to help in implantation, or they are not going to make the endometrium thicken. Instead, it will make it thin, so that even if fertilization happened, the implantation does not take place. And apart from that, they do one more thing, that is, after the vagina comes the cervix, right? So cervix is an entry for the uterus. So in there will be mucus present because of this hormone releasing IUDs. It makes mucus plug such that the sperm cannot enter through it. So hormone releasing IUD, you have progesterone. Do you think progesterone is the answer for this, or do you think LNG20 is the answer for this? Your actual answer is progesterone. Cannot be, because it is a hormone you're talking about. Yes, you make use of this hormone, that is different, but it is not a tool over here. What is a tool over here? LNG20. LNG20 or progestasert. If in the question, if in the option you have progestasert or LNG20, this would have been the answer. So hormone releasing IUD is this. The hormones, yes, it could be that, but we are not asked about the hormones. We are not asked about the hormones over here. Everywhere you're talking about what is a thing that you are using, right? So this is the thing that will be releasing hormones, and that is how it works. Now implants also have a similar function, that is, implants are put inside our body, okay, and beneath the skin and then what do? They are going to release certain hormones. So over here progestagens would suit better. Okay, over here it will be suiting better. So I'll say D goes with 2. D goes with 2 over here, and there's only one option like that. So I have to go for this option. Let me see if I got the answer correct. Let me bring that over here. It's too difficult to go there again and again. Come on. Yes, that becomes your answer. Okay, so LNG20, I remember it as LPG. I remember it has LPG. Either way, you have to remember. Okay, if you are anyways, you have to remember LNG20. And also remember one more thing, IUDs are best. First of all, it is widely accepted, most widely accepted contraceptive, and apart from that, basically it is best for spacing children. Okay, so for example, a couple had a child and now they do not want children until 5 years. So IUDs are best. So if in the question that comes, you have to tell it's IUD. Now going ahead with the next question, that is 11th question. Match list 1 with list 2. Lipase, nuclease, protease, amylase. Baba, easiest thing, we are going to spot the easiest one, protease. Protease, what they are going to do, they are going to break down the proteins. And in the proteins, what bond is there? Peptide bond is there. In the amino acid, proteins are made up of amino acid, and amino acid, they are bonded with each other through peptide bonds. So protease are going to target peptide. So I will say, I know this, so I am going to go with 1. Okay, C with 1. With that information, can I solve the answer? Let's see. So I have 2 options like that, with C is with 1. So I'll cancel this and this option. Cool. Now going with nuclease. Nucleic acid. Okay, nuclease, nucleic acid. And in nucleic acid, what do we have? We have DNA and RNA. In DNA, RNA, we know phosphodiester bond between the nucleotides. Yeah, between the nucleotides, what do we have? Phosphodiester bond. So let's make B with 4. B with 4. I have 2 options. I have 2 options. Over here, I see B is with 4. So this becomes the correct answer, but let's see the other thing, okay? Lipases, again, lipases for lipids. Lipases for lipids. In lipids, what bond is there, my dear children? In lipids, what bond do you find? Tell me. In lipids, I find ester bond. Okay, lipids are there with ester. So A goes with 2. And now you have amylase. Amylase is basically talking about something sugary, carbohydrate. So glycosidic bond is there between what? Basically between the sugar and the nitrogen base and things like that. Okay, so probably amylase will be going with 3. So D with 3, and this becomes your answer. But as I said, with the limited amount of information, still you can find the answer. Use the tricks. Okay, protease goes with peptide, nuclease with phosphodiester, amylase with glycosidic, and lipases with ester, and that's how we got our answer, that is this. Now see, lipases break down fats, that is lipid. Okay, ester bond is there. Nucleases break down nucleic acid, that is DNA and RNA. What do we have? Phosphodiester bond. Protease break down proteins, peptide bond. Amylase breaks down carbohydrates, glycosidic bond. Okay, works. Chalo. Next, given below are two statements, one is labelled as assertion A and the other is labelled as reason R. Now listen, whenever there is an assertion reason question, read it out very carefully. That is how they are going to put some mistakes in there. Okay, so assertion A says, breastfeeding during initial period of infant growth is recommended by doctors for bringing a healthy baby. It is correct. It is recommended by doctor for bringing a healthy baby. Cool. Now reason. Colostrum contains several antibodies absolutely essential to develop resistance for the newborn baby. Is it so? Is that why the doctors recommend mothers to breastfeed their children? Yes. It's because of that. The first milk which is there, it is yellowish in color, slightly yellowish in color. It is known as colostrum. And in which antibody is it rich in? IgA. A for Amma, you can remember like that. A for Amma. Okay, and Mama give you this thing. Fine. In that way, you can remember it. So first antibody to pass from mother to child is G. Is G. Always remember that. So we have 1, 2, 3, 4, 5 antibodies gamed. Okay, and this is the first guy to go from placenta to the baby. From the placenta to the baby. Second comes A, from milk to the baby. Okay, and MED does not have any sense. Okay, and one more thing I'll say. E. Okay, antibody E is produced during LH. Okay, that is given in your NCERT, at least know this thing. So I'll say assertion and reason both are true, and basically reason is the correct explanation of assertion. So can I say 1 is the answer? Yes, I can say 1 is the answer. Cool. Next, given below are some stages of human evolution. Arrange them in correct sequence, past to recent, as in from back to recent. Now this part of question, if you do not read, probably get things wrong. So first thing, listen to me carefully again. Okay, we had two organisms, that is Dryopithecus and Ramapithecus. Dryopithecus and Ramapithecus. Ramapithecus, as the name suggests, Rama, whatever, it's more human-like, ape-like. So Ramapithecus were the one that gave rise to Australopithecus. Okay, they gave rise to Australopithecus or Australopithecians, whatever you want to say. Then from Australopithecus, you got Homo habilis. The first ones to have proper human-like features, hominid features. Okay, you say yalla habibi, so you can remember like that, Homo habilis. Okay, after that, and always remember, these guys, they didn't have meat. They made tools and everything, but they didn't have meat. They essentially had vegetarian food. Okay, now came Homo erectus. Homo erectus, from this point you start having what? Meat and all. Now from Homo erectus came two things. One is Neanderthalensis and the second one is Homo sapiens. This came a bit late, this came a bit early. Okay, now you can arrange them in order. So basically we have Homo habilis, Homo neanderthalensis, Homo sapiens and Homo erectus. So over here I see Homo habilis is the first guy, A. Okay, and then we have what? Homo erectus, that is D. A and D. What option do you have? A and D. There's only one option. Okay. Now after that you have Homo neanderthalensis and then you have Homo sapiens, the latest. We are Homo sapiens, the latest version of human beings, right? So this becomes your answer. Cool. So Ramapithecus, Australopithecines, Homo habilis, Homo erectus, Homo neanderthalensis and then Homo sapiens. Works? Okay. Now if you suppose you have, just D. D was, C, that is, B, that is Homo sapiens, that we are Homo sapiens, you might be knowing about that at least, right? So with that option, can we try to find out anywhere where it ends with B, my dear children, I'll make sure that it might be, the answer might be between them. So it is going to become easy for me to try to find out the answer. So whatever you know, look for that in the option. It becomes more easy for you to eliminate the other options. Okay, works? Cool. Chalo. Sometimes, when I speak fast, I'm trying to speak fast because I want to, save as much as of time possible for you guys to revise certain things. I'm so sorry if it's going in high speed. You can lower it down the settings. I'll try to lower myself also down. Okay, and I mean my speed also down. And my dear children, please, if you are someone who were not able to solve the questions by your own, do not feel disheartened or demotivated. You still have time, okay? You are going to ace the examination. Just try to give your best, 100%. Just give your 100% and let's see what is going to be there as a result of it. But you have to give your 100%. Yes? Yeah. And yeah, that's all. Now, given below are some stages of human evolution. Array, we solved this, no? Again this question came up. Okay. Match list 1 with list 2. Axoneme, cartwheel pattern, crista, satellite. I know just one thing, ma'am. Crista. I have seen somewhere in mitochondria. So crista is basically found in mitochondria. You see in mitochondria you have this thing, right? The inward thing, this is known as crista in the inner mitochondrial membrane. So C goes with 4, and with that information, can I eliminate something? I got the answer actually with that. So C goes with 4, there's only one option like that. Let's see, rest of the options, okay, rest of the things. So A with 2, axoneme with 2. Yes, axoneme is basically a part of cilia and flagella. Okay, now cartwheel pattern, where do I find it? In the centrioles, so B goes with 1, yes. And satellite, you know satellite DNA. So at the DNA, some chromosome, now if you see, at the end of it you can find portion like this, which might be coding for nucleolus, okay, which might be coding for certain proteins and things like that, which is very essential for the formation of nucleolus. Inside the nucleus you have nucleolus, which is responsible for ribosomal unit formation and things like that, you know. So basically the satellite DNA is there at the tips, mostly towards the tip, and yeah, that's all. So satellite with chromosome, and with that you got the answer. If you know one thing, you got the entire answer. I'm telling you, this is of great help. So axoneme is the core structure of cilia and flagella. Okay, cartwheel pattern found in centriole, microtubule arrangement. If you see, it's a cartwheel pattern, and crista enfoldings of mitochondria, and then satellite, small chromosomal part seen in some chromosome, not all. Okay, cool. Now match list 1 with list 2. These are examples, purely examples, something that you have to by heart. Okay, chalo. I know again one very important thing, with that you can help yourself. So flying fish is a very common thing and it is kind of fascinating, which is why I tend to remember it. And flying fish is known as exocetus. Flying fish is known as exocetus. So D goes with 4. D goes with 4, but all the options are like that. So I can eliminate just this option. Cool. Now you have Pristis, you have Myxine, you have Pterophyllum. What do you say? What will be the answer for this? Sawfish. Okay, sawfish. Have you any idea about sawfish, my dear children? No? So let's see over here. Easy to remember. So Pterophyllum is angelfish. Okay, listen. It's angel. It's angel, but angel, if it all of a sudden you see an angel, my dear children, okay, all of a sudden when you are sleeping, when you open your eyes, you see someone out there, that's the angel, but you will be terrified, right? So Pterophyllum, because you will be terrified, and this is known as angelfish. Okay, so A goes with 3. Now Myxine. Myxine is basically hackfish. Is what? Hackfish. And Pristis, it is the sawfish. Okay, Pristis is what? Sawfish. You can see like it has some snot like this with things like that. You might have seen it. I don't know how to make it. It's like not like this, looks like a crocodile. Yeah, not like this, but yeah, something. Okay, I can try to make it once more. So it has some saw-like thing like this, okay, with which it moves. So this is how you can remember. So exocetus is flying fish, my dear children. Okay, and then Pterophyllum, you get terrified because you see an angel, and then Myxine is basically hackfish, and Pristis is what? Sawfish. So basically over here I've written one more thing. Pterophyllum is angelfish, a popular freshwater aquarium fish. Myxine is hackfish. It is jawless. It is jawless. That means it comes under cyclostomata. Cyclostomata means they have a circular mouth, and over here they do not have jaws. So their jaw looks like this. So I mean, your teeth and everything is like embedded in this particular round mouth like structure. They do not have a separate jaw like us. Okay, that is Myxine. And Pristis is sawfish, a cartilaginous fish. That means it comes under Chondrichthyes with a long saw-like snout. So Pristis is what? Cartilaginous, Chondrichthyes. Remember that. Exocetus is flying fish. I told you already. Now going ahead to the next thing. Match list 1 with list 2, and alpha antitrypsin, Cry1AB, Cry1AC, enzyme replacement therapy. So enzyme replacement therapy is given to a person who is suffering from a disease probably where the enzyme is not produced, and ADA deficiency is like that. ADA deficiency, it is deficient of some enzyme. right? Adenosine deaminase enzyme, that is what it is deficient of. So basically enzyme replacement therapy goes with 2, okay, it goes with 2. So D goes with 2 with that. Can I try to find the answer? Yes, again I can try to find the answer, and that is this. But let's see the other thing. Alpha-1 antitrypsin, it's a human protein. You know what? This human protein is used to treat one disease that is known as emphysema, which is a respiratory disorder that you have. Now, emphysema is that thing, and you make transgenic organism produce it, as in you basically put that gene into some other organism, and that organism produces this particular protein, and we take that protein to treat what? Emphysema. Because obviously we cannot make our body to do so again and again, and we cannot extract things, which is why we make other organisms do it, which is equally bad. Now, so you have got A with 3, yeah, that is correct. Now cry1AB, cry1AC, this thing I can make you remember it. Okay, so listen to me. Cry1AC and cry2AB, and then cry1AB. Now what is this cry? Basically there is a bacteria, Bacillus thuringiensis BT, yeah, and this bacteria has a gene that codes for toxins, that codes for what? Toxins. But in the bacteria's body it remains like that, inactive toxin. But when this bacteria is eaten up by something, for example, I am an insect, I came and ate a plant part which had this bacteria. Now this bacteria, when it reaches my mid-gut region, my dear children, what gut region? Mid-gut region. At that point of time, the toxin which is there, it becomes active, and all my epithelial lining cells, my dear children, they are going to be dead, and then I also will be dying. Okay, this is how it works. So cry1AC, and these genes can be taken from that bacteria and can be put into plant, so that any organism that comes and eat the plant could also die like this. Okay, now we have to see what are these genes, going to, or basically what set of genes are going to kill what set of organisms. Okay, so cry1AC and cry2AB, it is for cotton bollworms. Cotton bollworms. Ma'am, how do you remember it? So it's a very silly way to remember it, don't kill me. So first letter starts with C, so cotton. Second letter starts with B, so bollworm. Okay, now you might be thinking, ma'am, corn borers are also like that. But corn borers, if you say the small word, this is a big word. So big word needs big things. So 2 things you have for this. Okay, now cry1AB, so C over here and B over here, B over here, and with that we finish corn borers. So cry1AB is over here. There's only one thing for corn borers, okay, and there is 2 things for cotton bollworms, cry1AC and cry2AB. Okay, cool. So cry1AB is for what? Corn borers. Cry1AC is for cotton bollworms. If you have any more other tricks, you can tell me in the comment section. So this becomes your answer. Okay, cool, works? Yes. Chalo. Now, the TI plasmid of Agribacterium tumefaciens stands for TI plasmid. So, Agribacterium tumefaciens is a bacteria, okay, and it has a chromosome, it has its own chromosomal DNA and things like that. Apart from it, it has plasmid. It has plasmid, and you know the worst part of this plasmid is that it induces tumor. It causes tumor. So for example, I have a plant and this particular bacteria goes and affects this plant. What does it do? It basically transfers this TI plasmid in there. And what does this TI plasmid do? As the name suggests, it is tumor inducing, my dear children. But how can it do that? Basically, it has a region. It has a DNA which is known as tDNA or transfer DNA. What do you call it? Transfer DNA. Basically, when this DNA is transferred into this particular plant, what will happen? This particular plant will begin having what? Tumor in there. So TI stands for tumor inducing, and who is the one that induces? This transfer DNA, which is the tDNA. Cool. So your answer becomes this: tumor inhibiting, tumor independent, temperature independent, nothing. It's 3. Okay. Next, match list 1 with list 2. Again, chalo. It's the example based question. We have to do it. So pleurobrachia. Okay, pleurobrachia. How do I remember it? There's no way to remember it. You have to picturize it. So pleurobrachia basically comes at ctenophora, where you have 8 bands which are bioluminescent, which are very pretty and things like that. You have to remember like that. So pleurobrachia comes under ctenophora. So A goes with 2. A goes with 2. I have 2 options like that. Not this. not this. Okay, I have two options like that. Now radula is very specific feature. I can only find that in mollusks. In mollusk there is a radula, which is like a rasping thing that we have, and that is only found in what? Mollusk, remember. So B goes with 1. So B goes with 1. I just have one option like that. So this should be your answer, but let's see. Stomachode, yeah, this you can remember. So my dear children, we have 11 phyla, if I remember. Yes, 11 phyla. Last one is chordata. Rest all of them are non-chordates. Rest all of them are non-chordates. The 10th one which is there, 1, 2, 3, 4, 5, 6, now 10 is there, 11th is chordata, right? 10 is known as hemichordata. Why? Hemichordata. Initially it was considered to be under chordata, but now it is a separate thing, hemichordata, because it is like in between non-chordates and chordates. But now you put them under non-chordates only, but initially they were thought like that. Why, ma'am? Why was there actual confusion like that? Because chordates, they have a unique feature, that is to have a notochord. Chordates have a notochord, and hemichordates have something very similar to it, which is known as stomochord. Okay, so your C goes with hemichordata. C goes with 4. Cool. So C goes with 4. Yes. Now air bladder. Air bladder, where do you need air bladder? Only in the case of fishes. They are the ones, they have to be floating around like in the water, right? And that is found in Osteichthyes. That is bony fishes. In the case of bony fishes, you have air bladder. Chondrichthyes, do they have air bladder? No. Which is why they have to constantly swim around. Okay, they are cartilaginous and they have to constantly swim around. Osteichthyes, they need air bladder. Osteichthyes, they are made up of bones, right? So if they do not have air bladder, because of their weight only they will go down. Remember like that. Okay, you remember like that. That's not the actual logic, but yeah. So again, what do you got the answer as? You got the answer as 2. Okay. Okay, cool. So radula in mollusca. Now, which of the following is not a natural traditional contraceptive method? So natural, under natural comes 3 of them. Okay, natural means you are not using any tools, devices and things like that to prevent conception. For example, you have periodic abstinence. What do you mean by periodic abstinence? Right over here. It means that you do not have sex during the fertile period. That is basically around 10 to 17th day of the cycle, of the menstrual cycle. You do not have sexual intercourse. Why? Because this is the most fertile period. At this point of time, ovulation can happen. Egg can be released. If sperm also come, they can fertilize. So that is known as periodic abstinence. Okay, in this period you are absent. You are not doing anything. Now coitus interruptus. That means you are interrupting between the coitus. That means in between the sexual intercourse, there is an interruption happening. Why? It is also known as withdrawal method. It is also known as withdrawal method because before ejaculation, the penis has been retracted from the female body. That is known as coitus interruptus. Again, do you have any tool and anything like that used? No. So this is a natural thing. Now, lactational amenorrhea. What do you mean by that? When a mother is lactating, that means when a mother is breastfeeding at this point of time, okay, when she has milk in her body, at this point of time, usually effective for 6 months, okay, initial 6 months, she would not be menstruating. There won't be any menstrual cycle. That means there is no ovulation and things like that happening in her body. So at this point of time also, performing sex would not result in what? Having a child. But is it reliable? This is again a very important question. Is it effective? It is not at all effective. Not at all effective. There are higher chances of failure. Higher chances of failure. Okay, do not remember this, remember this. Do not remember this part, try to remember this part. What's higher chances of failure is there in the case of lactational amenorrhea. Amenorrhea means absence of what? Menstruation. That is what you mean by that. Now, so there is only one thing that is over here that is exception to it, and that will be walls. So walls are things that are acting like a physical barrier, okay, that does not allow the sperm to go inside the cervical region. So that is what you mean by walls. Cool? Cool. Next. Following are the stages of cell division. Okay, you have to choose the correct sequence of stages from the options given below. So, in cell division, my dear children, okay, we have interphase, which is there for 23 hours. If I say a cycle is of 24 hours, 23 hours a cell is going to spend in what? Interphase. Interphase. Okay, now 1 hour, ma'am, what does it do? It actually divides. So, I will call it M phase. I'll call it M phase. So your M phase is this much, rest all of your interphase. So in interphase, my dear children, you have gap 1 phase, you have S phase, and then you have gap 2 phase. Which one is the biggest one over here? It's gap 1 phase. Okay, what happens in gap 1 phase? Basically, over here, certain proteins are going to be made, some organelles, only some organelles are going to, duplicate, okay, because the cell is dividing, and suppose I am the cell, okay, and I am dividing into 2. Basically, both the other parts should be having equal amount of things, right? So duplication has to happen. So some organelles, they duplicate over here, some proteins are made over here, but majorly the cell growth takes place, cell grows in size, okay, that takes place over here. S phase is where DNA replication happens, okay, DNA replication, centriole duplication, all of this thing happens in the S phase, synthesis phase, okay. Now gap 2 phase is there, but the rest of the organelles which are not duplicated, they will duplicate, okay. Rest of the proteins which have not been formed, they will be formed, and things like that, which are very much important for cell division. And then you have the M phase. Now certain cells are there which do not want to enter the cell cycle at all. So they are in another phase which is known as G0 phase. Okay, quiescent stage or resting stage, whatever you want to say, it's over there in G0 stage. Now if you want to make in order, so I'll say gap, probably they might have meant gap 1 stage, so gap 1, E goes first, okay, E goes first, and then you have synthesis phase, so E then C, E then C, okay, it's found over here. So I think this is the answer, but let's see. E then C then gap 2, yes, until now it's clear. And now what do we have? Cytokinesis or karyokinesis? What do you mean by karyokinesis? Karyo means nucleus, kinesis means division. So nucleus division takes place first or cell division takes place first? Baba, everything is dividing into 2. So nucleus also which is inside the cell, there, it also has to divide into 2. Then only the cell will divide. So karyokinesis comes first in M phase, and then comes cytokinesis. Okay, so I'll say this comes first and then comes this. So D and then B, this becomes your answer. And in karyokinesis, you have the prophase, metaphase, anaphase, telophase, and things like that. Remember it. Cool, works. Children, we have completed 20 questions. That's a great deal. Are you enjoying the video? Do let me know in the comment section. Do share it with your friends as well if you are enjoying. Now, which one of the following factors will not affect the Hardy-Weinberg equilibrium? Is not going to affect, that is the question. Please remember the question, read the question carefully, okay? Now, Hardy-Weinberg law, what does it say? It says that basically the allelic frequency is going to remain constant generation after generation. That means, if you're not able to understand this big tough sentence, okay, so allelic frequency, for example, I have capital T in 5, small t in 3 amount. So this frequency of alleles is going to remain same for generation to generation. That is what the Hardy-Weinberg law says. But can it be true? No. Suppose some tall people came over here, again basically aren't you increasing the capital T amount? Some people left over here, again aren't you decreasing it? Okay, something happened which is why all the tall people got killed, some random thing, earthquake happened and tall people got killed somehow, randomly, random chance, or probably there was a tall unit association and a car came and crashed it, so all the tall people, they died off there. Okay, so that is known as genetic drift occurring by chance. Okay, gene migration, as I told you, some people going out, some people going in. Again, the genes are also migrating their body, and within the body they have the genes, they are also migrating. Now, genetic recombination, for example, over here, during sexual intercourse, obviously you have gametes being found. During the gametes being found, my dear children, there is high amount of recombination happening. So, new combination happens, which is why there is no capital T, there is no small t, there is something new arising out of it. Okay, again there is an allelic frequency change. All of it are going to change the allelic frequency. You are asked what is not going to affect the Hardy-Weinberg principle. So, genetic recombination is going to affect, genetic drift is going to affect, gene migration is going to affect. What is not going to affect? Constant gene pool. That is what is said in Hardy-Weinberg law, right? That it will be constant generation after generation, which is absolutely false. Okay, now if you are asked what are the factors that affect the Hardy-Weinberg principle, please remember genetic recombination, genetic drift, gene migration or sudden mutation or natural selection. Mutation, obviously it brings changes, and natural selection, oh I said mutation and naturation, oh my god, mutation, and I can write properly, I hope you know that, but it's just because it takes longer for me to write properly. So, natural selection and then mutation, these things are also there. Natural selection, for example, I took the small ones because the small ones could hide easily. So nature selected them. Things like that. Okay, so nature is selecting it. So in that way also their allelic frequency will increase, the other will decrease. So these are the things that you have to remember. Next, match list 1 with list 2. Okay, we have certain parts of brain over here and their functions as well. So the easiest for me to spot always is cerebellum. I'll tell you why. Cerebellum. Why? Cerebellum has, if you see the spelling, cerebellum, it has 2L, and 2L reminds me of the posture. I have to maintain straight posture. So cerebellum is a part of your brain that is responsible for your posture, your balance and everything. People who drink alcohol, when alcohol hits, okay, so it hits the cerebellum, which is why they walk like this. Okay, they are not able to have the balance. So cerebellum D goes with 1. Provides additional space for neurons, regulates posture and balance. This posture hit my eye at the first. So D goes with 1, and do I have an option with like that? One option like that. And I have one option like that. So this should be your answer, probable thing, but let's see. Now pons. What does the pons do? What does the pons do, my dear children? So pons is a part of your brainstem, pons, medulla oblongata and things like that. So pons is like for example, this is where you find your pons, okay, and basically that is something that connects different regions of your brain. So I will say connects different regions of the brain. Now medulla, very much important, again something that can catch my eyes very easily. Medulla is responsible for the rate of my heartbeat, for my respiration rate and for all the important things. Okay, medulla oblongata is very much important. There's a Malayalam movie, I'm a Malayali, okay, if anyone knows. Anyways, so there's a Malayalam movie in which the guy hits the medulla oblongata, okay, or there's some dialogue in there, and they says that person will immediately die off or things like that. Why? Because it controls, it has a very important role in that. Okay, in controlling that. Even though that was such a bad example, please remember it. Please do not remember the example. It's totally bad. Do not kill anyone. You have to be good children. Yeah. So we are not taught like that. Yeah. We have to be really good. No killing of even organisms. Okay. So controls respiration and gastric secretion. What is it? Medulla. So C goes with 2. Now hypothalamus. You're left with hypothalamus. Hypothalamus. What option do you have? You just have one option, that is neurosecretory cells. Is it so? Does the hypothalamus have neurosecretory cells? Yes, my dear children. And what do they do? Neurosecretory cells are the one that releases hormones. If you know, hypothalamus is the grandfather of the endocrine system. It is the one that commands the pituitary gland to release this and that. Obviously, it will be releasing hormones and saying that you have to release hormone, you have to release, do not have to. So hormones are the chemical messengers out there telling the other hormones whether it should be released or not. So who is the first guy to release it? It is the hypothalamus. Hypothalamus has neurosecretory cells which are going to release certain hormones. Okay, please remember it. So with that we get answer as 2. So hypothalamus were hormones. HH with the help of neurosecretory cells. Medulla, it's a very important thing, medulla oblongata, okay, controls respiration rate, heartbeat rate and things like that. Okay, cerebellum is for posture, remember that, and pons is basically connecting different regions of the brain. Yes. Now match list 1 with list 2. Chalo, expiratory capacity, functional residual capacity, vital capacity, inspiratory capacity. So over here I'll tell you one easy funda, okay. I don't think you will be able to remember it unless and until you know what does it mean. So which is why I have got something for you over here. I've not told you the answer though, okay. Expiratory capacity is the maximum amount of air a person can exhale after a normal inspiration. Inspiration as in, I took some breath inside air and now the maximum amount that I can leave out is known as expiratory capacity. Okay, so how do I find that over here? So probably expiratory capacity, I can say, is the normal breath. While during a normal breath, it's a tidal volume. It is known as tidal volume. Okay, and then expiratory reserve volume. This is the maximum that I can leave out. So tidal volume as in while normal breathing in, and then while normal breathing in, breathing out activity, I am also breathing out certain things like I release some air over there, right? Now, basically, if I am releasing all of it together, it is known as expiratory reserve volume. And that will give me what? Expiratory capacity. This is the capacity, the maximum amount of air a person can exhale after a normal inspiration. Now, you have functional residual capacity. What does that mean? The amount of air left in the lungs after a normal expiration. After a normal expiration, like I breathe out, whatever amount of oxygen is air is still there, it is known as functional residual capacity. So functional residual capacity, for that I have to calculate the residual volume. So expiratory reserve volume, that means the amount that I have given out, okay, plus the residual volume. Okay, plus what? The residual volume. It becomes what? Functional residual capacity. Okay, the amount of air left in the lungs after a normal expiration. After normal expiration, there will be some amount of air still left, that is known as residual volume. And expiratory reserve volume is what? The maximum amount of air a person can exhale. So after a maximum exhale, whatever is left behind over here is known as this. Functional residual capacity. Now vital capacity, the maximum amount of air a person can inhale and then exhale forcefully. Inhale and then exhale forcefully. So over here comes the tidal volume for sure, and also comes the expiratory reserve volume, the maximum I can give out and the maximum I can give in. So that is inspiratory reserve volume. All of it together will be what? The vital capacity. Okay, now inspiratory capacity is the maximum amount basically a person can inhale. So in that also again I will be having a tidal volume, that is a normal breathing, that, and apart from that the maximum amount, that is inspiratory reserve volume. So I will say this goes with, D goes with 3. I know it's a bit of confusing thing, it's not that easy to remember, but if you try you can. Okay, cool. So your answer should be A goes with 2, A goes with 2, all of them, no. There are two options where A goes with 2, this and this. B goes with 4, there is only one option, so this becomes your answer. C goes with 1, vital capacity goes with 1, and D goes with 3. This becomes your answer. Cool? Cool. Yes. Which of the following are autoimmune disorders? What do you mean by autoimmune disorders? That means what happens? Generally something foreign comes in, and that's how our body is built. Okay, our body will be killing that foreign particle. But over here in autoimmune disorder, our body is not able to differentiate between self cells and non-self cells. Okay, so basically it is going to target our own cell. Our own cells are getting damaged, that means we are going to have some problem in our body. Okay, so these are known as autoimmune disorder. In your NCERT class 12th textbook, okay, after allergy paragraph in human health and diseases, in a paragraph the example is given of what? Rheumatoid arthritis. But apart from that, if you also want to know more examples, what do we have? We have Myasthenia gravis, and also you have systemic lupus erythematosus. Okay, I always tend to mess it up. Yeah. So these three, A, B and E, these all are examples of what? Autoimmune disorders. Gout is caused when you have so much amount of uric acid being accumulated in your joints. Okay, that is how gout is being caused. Muscular dystrophy is basically totally genetic disorder that comes up. Cool. So your answer should be A, B and E. And do you have an option like that? Yes, we do have an option like that, which is why this becomes your answer. Cool. Works? Yes. Now I was just seeing the time. Yes. Next, which of the following is not a component of fallopian tube? Very easy question. Which of the thing is not a component of fallopian tube? So fallopian tube, if you have seen the hand-like structure over here, my dear children, you have fimbriae. Fimbriae is a part of infundibulum, and then comes ampulla, which is a broader region, and then comes a narrow region, which is not narrow over here because I'm very okay. Anyways, narrow over here, which is known as isthmus, Okay, so basically you have fimbriae, and then infundibulum, and then ampulla, which should be the broader region, and then isthmus is there, which is the narrow region. Where does fertilization take place? Where does the egg go and sit, my dear children? In the ampulla. Always remember, in the ampulla. That's where the sperm will be coming and fertilizing. Okay, now isthmus, infundibulum, ampulla, I spoke about that. What is uterine fundus? Uterine fundus is not a part of fallopian tube. If you have seen the structure of uterus, my dear children, in your NCERT textbook, there is something like this. Wait, just a second, okay? So there is, so here suppose you have the fallopian tube and things like that. Similarly here also you have it. This region is known as uterine fundus. This region is known as uterine fundus. So this is the part I was trying to show. It has narrow, but supposedly no one in normal human race do not have it narrow over here. We all have a broader region over here, okay, because we are strong. Yes. So this becomes your answer. Chalo. Match list 1 with list 2. Okay, it's about joints, fibrous joints, and then cartilaginous joint, and then hinge joint, and then ball and socket joint. Please understand that it's adjacent vertebrae, limited movement and things like that. So some things are coming over here, okay. So please go through that. Now I'll tell you one very important thing, okay. Always remember, it's easy to catch fibrous joints. What? Fibrous joints. You know what? Fibrous joints are found in over here, in the cranium, the skull bone. And the skull bone, can you do like this? Can you move around your bone? Okay, can it skip around? No, my dear children, they are not at all movable. Okay, they do not move at all. So fibrous joints is found in the skull, do not allow any movement. If it allows, it won't be really good. So A goes with 3, and I had just one option, A goes with 3, and with that I can spot. Trust me when I say I am here, coming here, and then trying to give you tricks. So it is occurring very naturally. So fibrous joints are easy, and that's how I try to find it out. You also can use these tricks, okay. No, if you're very sure about this, go for the easier way of choosing the correct answer. Now you have cartilaginous joint. This also I'm pretty sure. Cartilaginous joint are ones that will help in little amount of movement and things like that. Where do I find it? In the vertebral column. No, in between each vertebrae I have cartilage, which is why I am able to move like this and this and this, okay, majorly this. Okay, which is why I am able to move it. So cartilaginous joints are adjacent vertebrae with limited movement. With what? Limited movement. Always see this and this part of the answer should also be correct. So adjacent vertebrae with limited amount of movement. You cannot do all the things with that. You get it? Now talking about hinge joints. Hinge joints, have you seen the door hinges? Suppose this is a door, my dear. Closes, open, closes, open. Can it go back side? Can you do from here, this particular point of, can you put it back? No, you cannot do that. So hinge joint are found over here and also in your legs, in the knee joint. Okay. So hinge joint is found in the knee, helps in what? Locomotion. So hinge is like the hinge of the door. Now ball and socket joint. So one is like a ball and other would be like a socket. So this would be allowing me movement in like this direction and things like that. Where do I find it? I found it over here, and also in the pectoral girdle, that is over here in the hip, towards the hip region. Okay, so humerus, this is humerus, this is radius and ulna. Okay, so humerus over here in the shoulder you have a thing like this, and on this the bone is attached like this. So basically, which is why I am able to move like this. Okay, so ball and socket joints is towards humerus and also the pectoral girdle, and with that we found the answer, haven't we? So A goes with 3, yes ma'am, B goes with 1, yes ma'am, C goes with 4, yes ma'am, and D goes with 2. Cool? Works? Yes. Going ahead to the next thing. The flippers of the penguins and dolphins are the examples of what? Flippers of penguins and dolphins are examples of what, my dear children? Flippers. Okay, both of them have it. But dolphin is what? Dolphin is what? A mammal. Okay, penguin is what? A bird. Okay, but the function of flippers is to what? Swim. Swim. So, function is same. Okay, structurally if you see, structurally they are going to be different because do they share a common ancestry, as in very recent common ancestry? The answer is no. So ancestrally they are common, they do not have a shared common ancestry, recent one. Structurally they are very different, but functionally they are same. What are these structures known as? These structures are known as analogous organs. Analogous organs. And analogous organs show convergent evolution. Show what? Help in what? Convergent evolution. What is convergent evolution? Basically, two organisms are converging to a same point because they are staying in the same habitat, which is why they need same structures. That is convergent evolution. So always remember AC. AC, analogous organs in convergent evolution. So the answer becomes convergent evolution, not divergent evolution. Divergent evolution is where you have two different structures. For example, me and cheetah, we both have 4 limbs. Or you and cheetah, we all have 4 limbs, my dear children. Yeah, but I use it for eating, and that used for running and doing things like that. So if you see structurally we are all same. We have the radius, ulna and everything out there. Structurally we are same. We have the shared common ancestry and things like that. Because we all are vertebrates and we are mammals, right? But if you see functionally we are different, so that is divergent evolution. Divergent, why? Because we are in 2 separate habitats. I am over here in normal area, they would be different in a different habitat, feeding on different organism, which is why we have diverged, we have moved away. And these organs are known as homologous organs. So HD vision, you remember like that, homologous, divergent and AC for this. Okay, adaptive radiation is when one organism, the common ancestry, moves to different regions, giving rise to different organisms, but they have a common shared ancestry. So again, that is not the answer. Natural selection has no role to play over here. So yeah. Now 28th question, again there's an assertion and reason question. So you have to be really careful. As in statement questions, you have to be really careful. Statement 1 says, in the nephron, the descending limb of loop of Henle is impermeable to water and permeable to electrolytes. So if we have seen the nephron, so we have this, we have proximal convoluted tubule. This is the Bowman's capsule, proximal convoluted tubule, and then we have descending loop of Henle, and then we have ascending loop of Henle, and then we have the distal convoluted tubule, and then we have the collecting duct, right, collecting tube. So over here, the descending one, always remember this is bigger over here and goes down, so descending, that's why, and ascending because it is narrow over here and goes up, it becomes thick, so it is ascending. You can think like that. Descending one, it is always permeable to water. It will be permeable to water. It will let water ring because it is going thin, no? It is going thin, so it's like I need something, so let me be permeable to water. Okay, but over here it is impermeable to water. This area is the one that is impermeable to water. It looks like some anaconda ate some food, but yeah, for time being. It is impermeable to water, this is permeable to water. Now this is impermeable to electrolytes. Electrolytes cannot get in, electrolytes cannot move out. Over here, this is permeable to electrolytes, which is why it had some food and gained some weight over here. Okay, so it is permeable to what? Electrolytes. It is impermeable to water. There it is impermeable to electrolytes but permeable to water. It went on to a water diet. It went on to a water diet. You can remember like that. So here in nephron, descending limb of loop of Henle is impermeable to water. Wrong. It is permeable to water. Okay, it's on water diet. And permeable to electrolyte. Wrong, because this is impermeable to electrolyte. So statement 1 is anyways false. Let's see statement 2. The proximal convoluted tubule, that is this, is lined by simple columnar brush border epithelium and increases the surface area for reabsorption. So the only wrong part over here, it is not made up of simple columnar brush. Okay, columnar looks like this, but it's not columnar brush, it is basically cuboidal. Okay, this is what? Cuboidal cells, simple cuboidal cells. So again, this is wrong. Now if you are not paying attention to details, you will get this answer wrong as well. Okay, so can I say both statement 1 and 2 are false? Both statement 1 and 2 are false. This becomes your answer. Cool. Next thing. In both sexes of cockroach, a pair of jointed filamentous structures called anal cerci are present on what? 5th segment, 10th segment, 8th and 9th segment and 11th segment. They do not have 11 segments, so this answer is wrong. This option is wrong. Rest of the things. On the 5th, 8th and 9th, you do not have any things like that. On the 10th segment, you definitely have it, and that is known as anal cerci. It is found both in the male as well as the female. It looks like this. What is the use of it? Basically, they have certain hair-like structure, and that gives an idea of sensory hairs. Okay, that gives an idea of something coming up. For example, me going to kill the cockroach, that is why cockroach runs away, because it gets the senses on with the help of this hair, small hairs present on the anal cerci. Okay, so this is the thing. Okay, it's on the 10th segment. This is a direct question, you have to remember it. Now, which of the following factors are favorable for the formation of oxyhemoglobin in alveoli? Oxyhemoglobin, that means oxygen and hemoglobin are coming together. So what should be the condition? Obviously there should be high partial pressure of oxygen. Oxygen should be high in number, then only it can come and combine, right? If CO2 is high, obviously it will go and combine with CO2, even though it has hemoglobin has more affinity to oxygen. So definitely you need high PO2, that is partial pressure of oxygen, and apart from that, so do you have any other option like that? Here high PO2, here also high PO2, but here high PCO2 is also mentioned, that is becoming a wrong answer. Now high PO2 and lesser H plus concentration, is that so? Do you need that so? Yes, you need lesser H plus concentration. At this point of time only oxyhemoglobin can take place, can combine, can be formed. Okay, high PO2 and also lesser H plus concentration. So condition favorable for formation of oxyhemoglobin in alveoli are high PO2, less H plus concentration, low PCO2 and low temperature. These are the things that you definitely need it. Okay, and the opposite of it, more amount of H plus concentration if it is there, that means more amount of, let's say, PCO2 is also there. Okay, you can think like that. and that would not be favorable for this. So this becomes your answer. Next. The following diagram showing restriction sites in E. coli cloning vector PBR322, find the role of X and Y genes. So X and Y is over here. What is the role of this? So this is PBR, this is a plasmid, my dear children, and obviously this plasmid needs something which is known as origin of replication, ORI site. So X is the origin of replication, and what is the origin of replication? It obviously is the area from where the replication starts. So this is also an area which decides the copy number, how many copies of it can be made. So it decides the copy number, it is the area where it starts of the process of replication as well. Now Y is there. Y has a restriction enzyme site as well. What is Y? Y is ROP. ROP is a very important protein that is required for replication. Okay, replication. So not ROP is a protein. So ROP region is required for the process of formation of a protein, which is very much important for the process of replication to happen. So this is ROP site, and this is what ORI site that we have over here. Without ORI site, do you think all of this can work? Nope. Okay, so with the amount of information that we have, let's find out. The gene X is responsible for resistance to antibiotics. Wrong. That is only this gene and this gene. This is this is tetracycline resistant gene, and this is ampicillin resistant gene. Now Y is for protein. Y for protein involved in the replication of plasmid. This part of the answer is correct, but still wrong. The gene X is responsible for controlling the copy number of the linked DNA, and Y for protein involved in the replication of plasmid. Correct. Now gene X is for protein involved in replication of plasmid, and Y for resistance to antibiotics. Wrong. Gene X is responsible for recognition site, and Y is responsible for antibiotic resistance. Wrong. This is the answer for this particular question. Okay. Yes. Now match list 1 with list 2. Okay, fine. Down syndrome, thalassemia, alpha thalassemia, beta thalassemia, Klinefelter syndrome. So the thing that we know generally is Down syndrome. Down syndrome is basically when you have an extra 21st chromosome. Okay, extra 21st chromosome, that is trisomy of 21st chromosome, you say. So A goes with 3. A goes with 3. Do I have an option like that? I have just one option like that, and that gives me the answer. Now Klinefelter syndrome, that is found in, that is where you have certain problems in the sex chromosome, and over here you just have one of them, and that is X chromosome. So there is an additional X chromosome coming, which is why a male, it has supposed to be a male, but you have the feminine characters as well, because you are XXY. So this is again one addition, so trisomy again happening over here. Now alpha thalassemia and beta thalassemia, what does it show? So over here, let's see, C goes with 1. Beta thalassemia is on the 11th chromosome. So as in the beta globin gene, okay, mutation in it causes beta thalassemia. So beta globin gene, where is it located? It's on the 11th chromosome. Whereas alpha thalassemia, okay, alpha globin chain of hemoglobin is located on the 16th chromosome. Okay, please remember like that. So remember the opposite way. So B looks like this, 16 looks like this. Okay, so it's the opposite. Beta thalassemia is not on the 16th chromosome, it is instead on the 11th chromosome. Alpha thalassemia on the other hand, or you can remember like this, it looks like 16, right? The part of 6 like this, you can remember it. If you can, please remember it. Alpha thalassemia, alpha globin gene on the 16th chromosome, beta globin gene on the 11th chromosome. So this becomes your answer. The one that we targeted is absolutely correct. Okay. Now consider the following statement. Annelids are true coelomates. You have, okay, what do we have to find out? We have to find the correct answer. So whenever you have a question like this, do not read all the options and then read this. First read this, what you have to find out. That will save much of your time. So annelids are true coelomates. Are they? Do they have a true coelom? What do you mean by true coelom? Where basically you have 3 layers, ectoderm, endoderm, mesoderm, and in the mesoderm thing, okay, suppose this is endoderm, this is ectoderm, this is ectoderm, this is endoderm, and this should be mesoderm. This should be mesoderm, my dear children. Over here, they would be having a coelom, that is a space. Ayyo, I removed everything. Wait. What is the best way of explaining this? Wait. It happens. Okay. This is the endoderm. This is the ectoderm, and in this you have the mesoderm. Suppose you have a cavity in the mesoderm. You have a cavity in the, this is the cavity that you have in the mesoderm, and the cavity is lined by the mesodermal tissue right over here. The cavity is lined by the mesodermal tissue right over here. Are you able to understand or see anything? No, I think. So basically, anyways, supposing that there is a cavity, this white color thing that you see is a cavity, and basically when I am getting confused, so you don't have to follow it. So this particular cavity lined by mesoderm is known as coelom. And do annelids have it? Yes, annelids definitely have it. Annelids have true coelom. So this is the correct option that we have. Now, poriferans are pseudocoelomates. Arre baba, poriferans, they are made up of cells. They are what? Made up of cells. Okay, they not even have tissue thing and things like that. So do you expect them to have so many germ layers and things like that? No. So poriferans are acoelomates, do not have a coelom. Aschelminthus are acoelomates. No. Aschelminthus are pseudocoelomates. Are what? Pseudo. They have a false coelom. Okay, they have a false coelom. So they are not acoelomates, they are pseudocoelomates. Platyhelminthes are pseudocoelomates. Wrong. Platyhelminthes are acoelomates. Always remember. Okay, you have the first one to be poriferans, then you have cnidarians, then you have ctenophora, and then you have platyhelminthes. Right? Now, over here, these three guys are acoelomate because they do not have a proper structure only. They do not have that, this guy only have cells, these guys have tissues. Okay, so the germ layer formation and things like that becomes a lot more difficult. Okay, cnidarians and ctenophora, yeah, there is germ layers, but yeah, in poriferans you do not have it. But anyways, they are not very much complex, which is why they are naturally acoelomates. Talking about platyhelminthes, even though they have everything, still they are acoelomates. Still they are acoelomates. Okay, now after them come Aschelminthus, they would be having pseudocoelom, false coelom. Okay, and after that comes Annelida, Arthropoda and things like that. They are true coelomates. So your only option that is correct is this, that is A. So second answer becomes the best. Cool. Now match list 1 with list 2. Common cold, hemozoin, Vidal test and allergy. Very much easy. Very much easy. Why? Because Vidal test is found, there is only 2 tests mentioned in your NCERT. One is for HIV, that is ELISA, and the second one is for typhoid, Vidal. So Vidal test go with typhoid. So C goes with 2. C goes with 2, have 2 options like that. Chalo. Allergy, or allergy is even more easy for you. So D goes with 4. Dust mites causes allergy, my dear children. So D goes with 4. I have 2 options like that. Okay, let's go on. Common cold. Common cold is caused by a group of viruses which are known as rhinoviruses. And you know what? Viruses, which is why we are not. Do you have any medicine for common cold? Doctors say that after 7 days only it will be fine. Why? Because you do not have a drug being made for them. Viruses, they can change the genetic material every now and then. So even if I target one genetic material, they will change it all of a sudden. So I do not have a drug for that. So always remember, common cold is caused by viruses, and common cold does not affect your lower respiratory system, that is your lungs. Remember that. There's a big difference between pneumonia and common cold. Okay. Now hemozoin is produced in patients who have malaria. Malaria is caused by plasmodium. So, common cold goes with 3. So, B goes with 3. A goes with 3, and this becomes your answer. Even if I am not sure about hemozoin, still I get this as an answer because I am sure about common cold, which is caused by rhinoviruses. And hemozoin is a substance released by the cells effect. Okay, so plasmodium is there. Plasmodium goes and affects the RBC and breaks the hemoglobin into heme and globin. Heme is very toxic for the plasmodium, so it converts into hemozoin. Okay, which is toxic for us actually. Okay, so it safeguards itself for creating problem in our body. And when the RBC breaks open, hemozoin is released, which is why we get fevers and chills. Okay. Okay, cool. Now match list 1 with list 2. So we have a list of drugs over here, and we have list 2 as in what is their function. Now morphine is something that I want you to easily remember. Morphine is given by doctors. You should not be taking it. Okay, given by doctors for the people who have undergone surgeries, because it kills the painkiller, and it is also acting like a sedative so that they can easily sleep. So C goes with 1. Can I find the answer with that? No, because I have 2 options left, but I can eliminate 2 options. Okay, so this and this option is what we have to consider. Now morphine, when it has some chemical modification being done, okay, acetylation being done, you get something else which is known as heroin. So both of them you get it from a plant, that is opium poppy, or you also call them Papaver somniferum. Okay, so heroine, so you can remember in two ways. Papa is our hero, so Papa there is the one that gives, and basically Papa makes us sleep also when we have pain. Papa says, it's okay, it makes us sleep. So morphine also, you can remember in three ways. Basically they are the ones that is being produced. So I'll say heroine is 4. So B goes with 4, B goes with 4. I just have one option. This should be your answer. Okay, now cocaine, you get it from erythroxylum coca. You get it from erythroxylum coca. It is also known as coke or crack or things like that. Okay, now marijuana is basically you got it from cannabis sativa. From cannabis sativa, from different parts of it you get ganja, marijuana, hashish and things like that. So D goes with 2, A goes with 3. Cool, A goes with 3, D goes with 2, and this becomes your answer. Works? Chalo. Next. Given below are, we are starting section B, and section B, my dear children, in previous year you had an option of 15 questions. Out of them you have just had to answer 10, but this time you do not have any section B's like that, but anyway you have to practice the questions, right? So in total, if you have not seen that particular video about the change in exam pattern, go see it, okay, rather than practicing chumma. Now okay, chumma in Malayalam means, okay, or chumma in generally southern languages means do not waste your time doing nothing. Chumma as in nothing, yeah. So do not take it otherwise, okay. So that's what I meant. Given below are two statements. Statement 1, Gauss's competitive exclusion principle states that two close related species competing for different resources cannot exist indefinitely, coexist indefinitely. Do you think so? Gauss's competitive exclusion principle states that two closely related species competing for different resources. If they are competing for different resources, my dear children, why they cannot coexist? For example, you and me, if both of us want a teddy bear, we would be fighting for that. But if you want another teddy bear, I want another teddy bear, will we be both fighting? No. So this is wrong statement. Gauss's competitive exclusion principle states that two closely related species competing for the same resources cannot coexist indefinitely. So this is a wrong statement. Statement 2, according to Gauss's principle, during competition the inferior will be eliminated. During competition what will happen? The inferior will be eliminated. The same thing goes for your NEET examination as well. The person who have practiced all the questions properly, who have studied properly, they are the dominant ones, they will stay, but the inferior ones, they are going to get eliminated. Is this to scare you? No, this is for your self-awareness, that if you have still not given it a try of giving 100%, you should give it today. And my dear children, you all are going to dominate NEET. Okay, NEET is not going to dominate you, you are going to dominate NEET. Remember that, okay, have that in your mind. If you are in, do let me know, as I told you in the initial part of the session. If you are in, do let me know in the comment section that I am in for 180 by 180. Why I am asking you to comment? First thing, it gives you an affirmation that yes, I am going to do it. Second thing, I get to know that basically yes, my children have that belief that they are going to get it because of, so at least I might have helped in some other ways. That will definitely make me also happy, right, as a teacher. So definitely do that if you think so. Now, so according to Gauss's principle, during competition the inferior will be eliminated. This is correct. So I'll say statement 1 is false, statement 2 is true. This becomes your answer. Next question. The following are the statements about non-chordates. Non-chordates, okay, pharynx is perforated by gill slits. Okay, notochord is absent. Yes, notochord is absent. That is why they are known as non-chordates. Okay, central nervous system is dorsal. Is it? Heart is dorsal if present. Post-anal tail is absent. So these are the two features of chordates for general. Chordates, they definitely have what? Post-anal. They have the post-anal tail. So these guys are the one that would not be having it. So post-anal tail is absent, notochord is absent. So I'll say B and E there should be there in the option. So I just have one option like that where B and E is there. So this should be a correct answer. But let's see D. Heart is dorsal if present. No. Heart is dorsal if present. No. Heart is ventral. Do you think so? Do you think so? Is heart ventral? So see, there is a very important chart given over here in your NCERT itself, okay. So you have to, if you have to, check it out. So over here, chordates, notochord is present. Here non-chordates, notochord is absent. Central nervous system is dorsal, hollow and single. Central nervous system is ventral over here. It is towards the front side, not towards the dorsal side. Okay. Pharynx perforated by gill slits in us. Okay. But here gill slits are absent in us as in embryonic stages of us. Heart is ventral in us, but heart is dorsal if present. So over here, what is given? Over here, heart is dorsal if present. Is that true? Is that true? Yes, we have the heart towards the front region. They would be having towards the dorsal side. So this also becomes true. So if you are sure about chordates, okay, that notochord is, if you are sure that in chordates notochord is present, so here notochord will be absent. There if post-anal tail is present, here it will be absent. How can you remember? Chordates, as in us, my dear children, when we are embryonic, when we are in the embryonic stage, we definitely have a tail which goes off. Okay, which goes off when we become, let's say when we give, until the whole gastrulation is completed. At that point of time, gestation period is completed, my dear children, not gastrulation. Gestation period is completed, the tail and everything goes off. We definitely have gill slits also, that also goes off. Okay, so please remember that. So, and heart is dorsal if present, that also becomes the correct point. So, your B, D, E option becomes the correct answer for this. If you want to have a look at this chart, please do that. It's very, very important for you to know it. Cool? Cool. Next. Question number, okay. Question number 38. Given below are two statements, again statement, okay. The cerebral hemispheres are connected by nerve tract known as corpus callosum. Do you think the cerebral hemisphere, you have two hemispheres right over here, right? Are they connected with the help of nerve tract called corpus callosum? Yes, the answer is yes, this is true. Callosum, not callosum, yeah. Now, statement 2. The brain stem consists of the medulla oblongata, pons and cerebrum. Okay, the brain stem which is there, does it consist of pons, medulla oblongata and the cerebrum? No, cerebrum is this big area that we have, my dear children. We talk about the brain, this big thing that you have is the cerebrum. Okay, towards the end you will be having the cerebellum and things like that. Over here you would be having your pons, no, you will be having a midbrain, yeah, and then you have your pons and then you have your medulla oblongata over here. This becomes your brain stem. So your midbrain, your pons and medulla oblongata becomes your, what say, the brain stem. Not the cerebrum, neither the cerebellum. Cerebrum is a part of your forebrain. Okay, cerebellum is a part of your hindbrain. Okay, and pons and medulla oblongata is also part of your hindbrain. This is the midbrain. Okay, I'll just color it a bit. This is your midbrain, and this is your hindbrain, cerebellum, pons and medulla oblongata, and this whole thing forms your, what's a forebrain. Your forebrain consists of your thalamus, hypothalamus and then cerebrum and things like that. So over here cerebrum is mentioned, that is why it is wrong. The brain stem consists of medulla oblongata, pons and cerebellum. Che, and what, midbrain, not the cerebrum or even cerebellum. So this becomes your wrong statement. Only this part can become the brain stem. So I will say statement 1 is correct, statement 2 is false. Statement 1 is correct, statement 2 is incorrect. Cool? Cool. Right, yeah. Now, question number 9. Match list 1 with list 2 related to digestive system of cockroach. So, cockroach, we will study about cockroach. The structures used for storing of food. The structures used for storing of food is what? Just give me a second, okay? Just give me a quick second. Yes. So over here, the structure used for grinding of, the structure used for storing of food will be what? Crop. Yeah, and grinding. For grinding, what do we have? We have gizzard. So A goes with 4, crop, and D goes with 1, gizzard. A goes with 4 and D goes with 1. A goes with 4 and D goes with 1. I have 2 options like that. Let me eliminate the other 2 options. Okay. Now, ring of 6 to 8 blind tubules at the junction of foregut and midgut. What do you call this? This is known as gastric ceca itself. And ring of 100 to 150 yellow colored in thin filaments at the junction of midgut and hindgut is known as Malpighian tubules, which are very much important in the process of excretion. Okay, in the case of arthropods. So I will say C goes with 3. C goes with 3 and B goes with 2. A should be, the first one should be your answer. Yes, the first one is your answer. Gizzard for grinding. Okay, so storing we have crop, and 100 to 150 filaments, yellow thin filaments is what? Malpighian tubules for excretion. Ring of 6 to 8 blind tubules is gastric ceca. Okay. Now going ahead, match list 1 with list 2. So these are certain disorders that we have, okay, and then we have to find it with this one. So exophthalmic goiter. So goiter is caused when, my dear children, either there is a deficiency of thyroid hormones, okay, basically when iodine is not there, when iodine is not there, thyroid hormones have a problem because thyroid hormones are made up of iodine, okay, T4 and T3, right. Now exophthalmic goiter is caused when there is an excess hypersecretion of thyroid hormone, and protruding eyeballs are also going to be seen over here in the case of exophthalmic goiter. Okay, or basically in exophthalmic goiter, I mean that there will be a hypersecretion of the thyroid hormones and also there will be protrusion of the eyeballs. Okay, now you have what? We have Cushing's disease, okay, Cushing's syndrome actually. Cushing's syndrome is basically where you have excess secretion of cortisol, there is a moon phase and hyperglycemia. Hyperglycemia as in sugar content increases way too much. Okay, this is what Cushing's syndrome. So C for Cushing's syndrome, C for cortisol, that is how I remember it very easily. So Cushing's syndrome, excess secretion of cortisol, moon phase and hyperglycemia. What are we left with? Acromegaly and cretinism. What do you mean by that? So let's try to find the answer with this option that we have already in hand. Okay, so A goes with 3, A goes with 3, A goes with 3. I have 2 options like that. Okay, and then C goes with 1, C goes with 1, I just have one option with that, so this should be your answer. So listen, acromegaly is where you have excessive secretion of growth hormone. Okay, you would be having excessive secretion of growth hormone. Now cretinism is where basically hyposecretion of thyroid hormone and there will be stunted growth, exact opposite of it. Cretinism, stunted growth. Acromegaly, where there is an excessive secretion of growth hormone, which is why the height will be way too much. Okay, so excessive secretion of growth hormone in acromegaly, and cretinism is where hyposecretion of thyroid hormone is there and it will lead to what? Stunted growth as well. Okay, so 4 becomes your answer for this question. Why I have not targeted it? Yes, I have marked it. Cool, works. So did you understand until 40 questions? So we have 10 more questions, but I'll recommend you, okay, if you go back to, what say, if you complete the whole video and then go back to the things that you have learned, it might be difficult for you. So I'll request you take a break. 40 minutes are done, not 40 minutes exactly, more than that actually. So 40 questions are done, take a break, revisit all the questions just like that, okay, just to see the answers so that you have that in your head to process, and then go ahead with this 10 more questions. So you'll feel I just have 10 more questions to do. That is why there's a long pause. Works? So do that and come back again. I'll be coming back with the 10 questions right over here. Do not go anywhere, just go through it. Okay? So children, I hope you're done with the 40 questions revision, and now we are here with the 41st one. Let's begin it. Yes. So choose the correct statement given below regarding juxtamedullary nephron. Ma'am, what is that? So basically it's there in our kidney. Yeah, we have 2 types of nephrons majorly, that is cortical nephrons and juxtamedullary nephron. Okay. Now which is in more amount? Cortical nephrons are found in more amount. Almost 80% of them are cortical nephrons. Yeah. Rest 15 to 20 is juxtamedullary nephron. So which has more amount? Cortical nephrons. Now where are they found? Where are both of them found? Basically, if you see the kidney, we have 2 areas. The outer area is the renal cortex and the inner area is the renal medulla. So as the name suggests, cortical nephrons are towards the cortex. Okay. They mostly in the cortex. The loop of Henle basically dives into the medulla, but not very much. Okay, it's very little of the part of the cortical nephron that gets into the medulla. But when you talk about juxtamedullary nephron, basically it has a very long loop of Henle that gets into the medullary region, which is why it has got juxtamedullary nephron name. Now, juxtamedullary nephrons are located in the columns of Bertini. What do you mean by that? So suppose there is renal cortex, okay, and there is medulla. So inside the medulla also it is divided into different regions. So who does that? From the cortex extends certain things which basically dives down, and this is known as column of Bertini. I will be showing you the picture. These are known as columns of Bertini, and in between I told you there are certain things, right? Dividing them are these columns. So these are known as pyramids, and in these pyramids are our nephrons being located. It's not in the columns of Bertini, so I'll say this particular statement would be a wrong statement. This would be wrong. Now the renal corpuscle of juxtamedullary nephron lies in the outer portion of the renal medulla. Do you think renal corpuscle as in, what do you mean by that? Renal corpuscle as in, you're talking about Bowman's capsule and glomerulus and things like that, okay, that upper part of the nephron. So it is said that renal corpuscle of juxtamedullary nephron lies in the outer portion of the renal medulla. Is it in the outer portion of the renal medulla? No, it's in the cortex region itself. Yeah, it's outside the renal medulla, that is there, but it's not, you should not be giving reference to renal medulla there. You can just say it's near to the renal medulla if you want to, but majorly it is what? In the cortex region. Loop of Henle extends down in the medulla, but rest of the part is there in the cortex itself. Yeah. Now, this is also wrong. Now, loop of Henle of juxtamedullary nephron runs deep into the medulla. Yes, this is correct. Now, juxtamedullary nephrons outnumber the cortical nephrons. Do they outnumber? That means they are in more amount. Are they in more amount? No. I told you it's the cortical nephrons which are in more amount. Again, this is also wrong. What we had to find out? We had to find out the correct one, and here it goes. So if you're someone who's not still understanding the picturization of it, listen to me carefully. This is the cortex, and this thing that you see, the innermost the region over here, this is the medulla. Now as you can see, the cortex is dividing them. They are known as renal pyramids, okay, and in between you have seen columns. What is this column? This column is column of Bertini. So our cortical nephrons are basically towards this side in the cortex only. Little of the loop of Henle gets into the medulla. But in the case of juxtamedullary nephron, what they do is that their renal corpuscle is over here, but the loop of Henle goes deep, very deep, my dear children. That is the thing about them. And it is not found elsewhere, not in the columns, not found elsewhere, just here in the renal pyramid that you are having over there. Okay, so this becomes the correct answer. Now, going ahead with the 42nd question. Regarding catalytic cycle of an enzyme action, select the correct sequential steps. So, what happens during the catalytic action? Okay, so first thing that we want is enzyme. And at enzymes, what side does the substrate come and bind? At the active site. So if you want to have a catalytic enzyme action, my dear children, what do we need to do? We need to find the end substrate binding to the active site. This could be the first point. But can you come to this directly? No, you have to read down all the points. Substrate enzyme complex formation comes later. Free enzyme ready to bind with another substrate, that is next round. Release of products, array, first of all it has to bind. Chemical bonds of the substrate broken. So we are stuck with this substrate binding to active site. First it comes and bind to the active site. Then what happens? Basically, can I say substrate enzyme complex formation occurs? Yes. So E comes and then comes A. E comes and then comes A. So over here I just have one option like that. But let's see. Next. After that, substrate enzyme complex formation occurs, and then the enzyme would be acting on the substrate and breaking its own bond. Yeah, it will be breaking the bonds of the substrate, right? So chemical bonds of the substrate broken. This will be the next part. Yeah, and then after that, what will be there? Release of products. After the chemical bonds of the substrate have been broken, it is basically converted into what? The product. And this is being released. So release of product, and then free enzyme ready to bind with another substrate. What happens? Now we have the free enzyme which is ready to bind with the next substrate which is there. So can I say EADCB is the answer? Yes. Cool. And the same cycle repeats again and again and again and again. Fine? Good. Now, match the list 1 with list 2. Okay, match list 1 with list 2. So, what do we have? Unicellular glandular epithelium, compound epithelium, multicellular glandular epithelium, endocrine glandular epithelium. So, I can be sure of this one thing, that is D. Endocrine glandular epithelium, that means it is going to release certain hormones. And which among them are the ones that releases hormones? That's the pancreas. So, I will say D, go with 2. D, go with 2, I have 2 options like that, rest of them I can exclude. Okay, now I have what? I have unicellular glandular epithelium. Where do you find unicellular? That means each cell over there is acting like a gland. Where do you find that? In the goblet cells of alimentary canal. What do the goblet cells do? They release mucus, my dear children. So each cell over there is acting like a gland. So unicellular glandular epithelium is found in the goblet cells of alimentary canal. Alimentary canal as a whole digestive tract that is there, my dear children. You can find goblet cell in the stomach also, in your esophagus also, things like that, because you need the mucus lining, right? Yes. So I'll say A goes with 3. A goes with 3. I just have one option like that. This should be your answer. Now B. Compound epithelium, it goes with moist surface of buccal cavity. So it is more towards the area where you need some moisture and things like that. So compound epithelium is towards the moist surface of buccal cavity. We are left with multicellular glandular epithelium. That means multiple cells are there and they are going to act like glands. Where do you find it? In salivary glands. In salivary glands. Multicellular glandular epithelium in salivary glands. And with that we get the answer. So just to summarize, endocrine glandular epithelium in the pancreas, because endocrine as in it releases hormones, yeah. And then unicellular glandular epithelium in the goblet cells of alimentary canal, compound epithelium, moist surface of buccal cavity, and multicellular glandular epithelium, salivary glands. Good? Yes. Next one, yeah. So this is given over here. Unicellular glandular epithelium, these are single cell glands found in the digestive and respiratory tract that secrete mucus. Okay, now compound epithelium, moist surface of buccal cavity, provides protection and is found in areas exposed to what? Mechanical stresses, which is why this is there in the buccal cavity. Now multicellular glandular epithelium, these glands contain multiple cells and are involved in secretion like salivary glands producing saliva. And endocrine glandular epithelium, pancreas. The pancreas has endocrine cells that secrete hormones like insulin and glucagon in the bloodstream. That's all. Now again, match list 1 with list 2. So this is more towards evolution. Let's see who all can do it. So Mesozoic era, Proterozoic era, Cenozoic era, Paleozoic era. So I would like to go ahead with Proterozoic era. Pro means primitive, old. So among them, what are the oldest thing that we probably have? What do you think? Lower invertebrates. Invertebrates came, then came invertebrates came, and then came vertebrates and things like that. So we can go back to primitive time where we have got lower invertebrates over here. So B goes with 1. Where is the option where B goes with 1? All the options are there where B goes with 1 except this one. So I'll cut this off. At least I have got one option down. Yeah. Now talking about Mesozoic era. This is pretty much famous because in Mesozoic era, my dear children, you have the Cretaceous period, you have the Jurassic period and things like that. Mesozoic era, okay. And in Jurassic period, what did you have? You had the the dinosaurs and things like that. So can I say birds and reptiles are more towards the Mesozoic era? Can I say that? Yes, I can say that. So A goes with 3. A goes with 3. How many options do I have? I have 2 options. So let me cut this option also. I have these 2 options now. Okay. Now Cenozoic era is where you find mammals. Okay, Cenozoic era is where you find mammals. So C goes with 4. Cool. So this becomes your answer. And Paleozoic era goes with fishes and amphibia. So in that way, you get the answer. Always remember, Mesozoic era is where you have Jurassic period, Triassic period and things like that. So it's where you have the reptiles and things. Dinosaurs were kind of very dominant over there. Yeah. So birds and reptiles were dominant in the Mesozoic era, and then came Proterozoic era where lower invertebrates were there. Cenozoic era where mammals were there. Yeah. And mammals were seen. You can remember like that. And Paleozoic era is basically where fishes and amphibians rule the world. Okay. Cool. Now, given below are 2 statements. Mitochondria and chloroplast are both double membrane bound organelles. Are they both double membrane? Do they have double membranes? Yes, both of them, they are double membranes. So I'll say this is correct. Now, inner membrane of mitochondria is relatively less permeable as compared to chloroplast. Is that so? Inner membrane of mitochondria is relatively less permeable as compared to chloroplast. Actually, it's the opposite. Chloroplast, the inner membrane, is not permeable to a lot of things. Okay, it's in respect to that, mitochondria is kind of permeable. Little permeable, but yes, permeable to certain ions and things like that. So it should be the opposite way. It's the wrong statement that is given over here. Inner membrane of chloroplast is relatively less permeable, or you can say not at all permeable as compared to what? Mitochondria. That would have been more correct. So this is a wrong statement. So I'll say statement 1 is correct, statement 2 is incorrect. So I have this option. Cool? Yes. Now, match list 1 with the list 2. Okay. P wave, QRS complex, T wave and TP gap. What does that mean? So basically we have Chotusa, P wave right over there. Okay. What does P wave suggest? That the atrium which is there, okay, it is undergoing contraction. What do you call this? Depolarization of atria, my dear children. What is happening? Depolarization of atria is happening, which is why it is going to undergo contraction. Okay, this is the P wave. And now you have QRS right over there, QRS. Okay, this shows, okay, after atrium has contracted. It has sent the blood to what? Ventricles. Now it's whose turn to contract? It's ventricles time to contract. And when will this happen? When depolarization of ventricles have happened. So depolarization of ventricles go with QRX, okay, QRS, and P goes with depolarization of this. So P goes with 3, A goes with 3, A goes with 3. I just have one option. This should be giving me the whole answer as well. Okay, and QRS means depolarization of ventricles, yes. And now comes, there is a T wave. There's what? A T wave. What does T wave suggest? T wave suggests that basically what is happening? In T wave, repolarization of ventricles are happening. That means they are going to start again. They are going to, come back to this thing that is relaxed mode. Okay, ventricular relaxation will be happening in T wave. And what's the TP gap? From T to P. What do you call this gap as? What is happening over here? Heart muscles are electrically silent, as in both of them they are going to have a joint diastole period. Both of them are going to relax. Okay, in this particular point of time the atrium will be filled with blood. Okay, in this relaxation the atrium will be filled with blood. So I'll say TP gap denotes for this. So hence your answer will be 2. So A goes with 3, depolarization of atria, QRS with depolarization of ventricles, and then C with repolarization of ventricles, and D with heart muscles are electrically silent. And this becomes your answer. Next, given below are 2 statements. Statement 1, bone marrow is the main lymphoid organ where all blood cells including lymphocytes are produced. Are they? Yes, bone marrow is an area where all the cells, my dear children, it is where you have hematopoietic stem cells, that means blood stem cells, which will be giving rise to different type of blood cells out there. Okay, whether it be lymphocytes or basically, white blood cells or basically red blood cells or platelets, whatever, they are going to be formed over here. Now statement 2 says both bone marrow and thymus provide microenvironments for the development and maturation of T lymphocytes. Is it? So yes, development of T lymphocytes occurs in the bone marrow, maturation occurs in the thymus. So thymus is not an area where it is produced or developed. Okay, remember that. Thymus is just for maturation of T lymphocytes and also for making it antigen sensitive, so that it knows that hey, you whenever something foreign comes, you have to kill it. It becomes antigen sensitive, does not work over there, does not kill anything over there. It's just becoming antigen sensitive. So maturation of T lymphocytes occurs in the thymus, whereas development occurs definitely in the bone marrow also. Now, B lymphocytes, what about them? They mature here itself, in the bone marrow itself. So I'll say both the statements are equally correct. Yeah, both the statements would be correct, even though not politically entirely correct, but still. So both bone marrow and thymus provide microenvironments for development and maturation of T lymphocytes. Okay, cool. So I'll say both statement 1 and 2 are correct. This should be your answer. Yes. Now, identify the correct option, that is A, B, C and D with respect to spermatogenesis. Okay, so basically J and RH is given over here. What is that? I told you, you have hypothalamus secreting a hormone, that is gonadotropin releasing hormone. Hypothalamus is the grandfather of the endocrine system. Yeah, it releases gonadotropin releasing hormone. What are gonadotropins? LH and FSH. LH and FSH, they are gonadotropin. So it is saying it is releasing gonadotropin releasing hormone. Okay, it will go and act on, this gonadotropin releasing hormone will go and act on pituitary. Which part of pituitary, my dear children? Anterior part of pituitary, and make the pituitary release 2 things. One is FSH and the other one is FSH, other one is LH. What will happen to the LH? LH will be going and acting on L for LH, luteinizing hormone, and L for Leydig cells. It will be going and acting on the Leydig cells, and the Leydig cells will be releasing androgens, and these androgens will be helping in the formation of spermatids out there. Now FSH, what do they do? Where do they go and act? FSH, S stands for Sertoli cells. So it will go and act on Sertoli cells, release some factors and help in what? Release some factors and help in what? There is a thing, okay, in spermatogenesis formation, at the end you get spermatids, and spermatids are basically transformed into spermatozoas, a more mature form. Okay, this is known as spermiogenesis. What do you call this? Spermiogenesis. And here these factors are going to play a very important role. So I'll say spermiogenesis is over here. What is spermiogenesis? Listen to me carefully, okay. So there is spermatogonia. It differentiates, divides, okay, divides and differentiates into one cell will be there. This is known as primary spermatocyte. Now this will undergo meiosis into 2 cells, secondary spermatocyte. At the end again 2 cells. What are these? These are spermatids. spermatids. They have to be getting matured and they are like round globular in shape. But sperms, are they round or they elongated? They are elongated. So you transform them in elongated form, which is known as spermatozoa. What do you call this process as? You call this process as spermiogenesis. Spermiogenesis. And for this you definitely require a lot of testosterone. So the testosterone have to stay in this particular place. So who will hold this? It will be held by the factors released by the Sertoli cells, androgen binding proteins. That is what you call it as. Okay, so they will bind the androgens right over here and help in the process of spermiogenesis to happen. Listen to me carefully. Spermiogenesis, especially spermiogenesis. This whole process is spermatogenesis. I am talking about spermiogenesis. For this particular process you need these factors so that androgens can stay at one place and help. Okay, remember that. All of the place you obviously need testosterone. I am not saying it is not needed, but over here you definitely need it, and that's why these factors are being released as well. So what is A over here? A is FSH. Yeah. What is B over here? B are Leydig cells. Okay, what is C over here? C are Sertoli cells and spermiogenesis. This becomes your answer. I don't have to look forward the next options as well. Cool. And later when the spermatozoa moved out of the what do you call seminiferous tubules, what do you call this? This is happening in the seminiferous tubules, right? When they move out, what do you call it? You call this spermiation. Aviation, spermiation, remember, like that. Okay, just from the seminiferous tubules it is moving into the what do you call rete testis and tubuli recti, all these things that is up there. Okay, into the accessory ducts. Now, as per ABO blood grouping system, the blood group of father is B positive and mother is A positive and the child is O positive. The respective genotype can be. So if I am saying the child is O positive, okay, then the child should be receiving 2 II gene from both the parents. 1 I from here, 1 I from there. So this I has become O somehow. Yeah. So basically one parent should be having 1 I, another parent should also be having 1 I, but the parents are having B and A blood group. So what do I say? It might be like this, IB and IA. You get it? Then only you can receive, no? Get it? So what is father's blood group? It will be IBI. Mother's blood group, it's be IAI, and the child's blood group will be II. Okay? Fine? Cool? So can I say A is the correct answer? Or do you have any other option? No, I do not have any other option, my dear children. If there is IB over here, so B and I cannot come together. You need O blood group, and for O blood group you need this, because O is the recessive group out there. Okay, so you need 2 homozygous II. So choose the most appropriate answer from the given option. I will say A only. This would be the answer. Yes. And now match list 1 with list 2. What is list 2? Okay, we have RNA polymerase 3, termination, transcription, splicing of hormones, starter box. So easy. Let's go easy. Okay, so RNA polymerase, while it completes transcription, while it's time to say bye-bye to the DNA, because it has completed transcription, it needs a factor to come in, and it cries for that factor, like, please help me, I don't want to be here. So it's Rho factor. Okay, what do you call it? Rho factor. The termination of transcription occurs when RNA polymerase combines with the Rho factor. Okay, so B goes with 3. B goes with 3. I just have one option, and with that I can get the answer. Okay, and for starting off transcription, what do I need? I need sigma factor, and for letting it go, Rho factor. Okay, remember that. Now RNA polymerase 3, it is responsible for the transcription of, or basically formation of, snRNAs and tRNA. This tRNA comes forward, that means you have to easily guess it out. So we have 3 types of RNA polymerase, 1, 2 and 3. First one is the one that helps in the formation of rRNA. Second one that helps in the formation of hnRNA, heterogeneous nuclear RNA, and also mRNA, the main transcription thing is done by RNA polymerase 2. Okay, and then we have tRNA and snRNA all being formed by RNA polymerase 3. This is in the case of eukaryotes. Okay, so yes. So now splicing of exons. Splicing of exons, basically that means you are removing the introns and you are joining the exons. Okay, so it should be splicing of introns though, but anyways, who does it? It is snRNPs. Okay, so snRNPs becomes a part of spliceosomes. Spliceosomes are big molecular machinery, my dear children, which will be involved in the cutting of introns and removal of that. Okay, in the process of splicing. We do not need introns. Do we need introns in the DNA or in the hnRNA? DNA also has it. hnRNA RNA also has it. There are regions that codes for proteins, there are regions that does not code for proteins, and that are introns. These introns have to be removed and the exons have to be joined. For the next process, that is what? Translation. Because you just need exons to code for proteins, that's why you're removing this. This process is known as splicing. And with the help of whom is it done? SNRNPs. The other one which will be helping small nuclear RNPs, I forgot the full form, yeah. So they are the one that will be removing it, okay. So splicing of exons will be this. Now Tata box, so you might be thinking it might be at the end, that's why it's not a Tata box. No, it's in the beginning, it's in the promoter region, okay, quite near to the promoter region. Leaving few base pairs, we have the Tata box, that means it has a sequence of TA, TAA, things like that. What does it suggest? That hey polymerase, you have to come and attach over here. Hey RNA polymerase, you have to come and attach over here. This is where you will find the promoter region and things like that. So it is there for helping in what? Initiation of transcription to happen. So it's more towards the promoter. So instead of saying hi box, it is Tata box. This is a contradictory thing, but yes. So D goes with 2. And that we solve all the questions out there. Yes, all the 50 questions are being completed for NEET Zoology 2024. How do you feel? I'll tell you something very honestly. It's not like the exam is too difficult. No, I think it was easy to moderate. It is not very much difficult. I didn't find too difficult questions out there. If you are someone who have read through your NCERT properly, if you are someone who have solved enough questions, I think it will be really easy for you. So rather than listening to 10,000 people who would be suggesting you so many books and things like that, which you obviously cannot refer to as of now, I'll recommend you to stick to your NCERT, okay, and practice as many as questions as you can. That will be really helpful for you guys. Yes, and also listen to these videos where the concepts are being cleared. It will help you out really well. And I just wanted to tell you, thank you for watching this video. Do not forget to like, share and subscribe, and also comment down your valuable things. If you have any doubts still up there, please ask me. I'm here to help you out. Yes, and definitely recommend it to your friends as well. Bye bye, take care, wishing you all the best. You guys are going to do so good. Bye bye.